Approximately 260 million years ago, Earth supported an ecosystem of a complexity and diversity that would not be matched again for tens of millions of years. The atmosphere carried more oxygen than the planet holds today, concentrations approaching 30% by volume, compared to the modern figure of roughly 21%. That difference, sustained across geological time, produced consequences measured not in degrees but in fundamental biological possibility. Life, given that abundance, expanded into forms and scales that stretch the limits of what the term animal is typically understood to mean. The land masses of the late Permian period had not yet separated into the continents recognized today. They were joined into the supercontinent Pangaea, a single continuous body of land stretching from pole to pole, bordered on all sides by the vast global ocean known as Panthalassa, which covered approximately 70% of the planet's surface. In the interior of this landmass, far from any moderating coastal influence, climates swung between extremes: hot, dry summers inland, cold winters at high latitudes. But along the margins and in the low equatorial zones, conditions were humid, warm, and biologically productive on a scale difficult to overstate. The forests of this period bore little resemblance to those of the modern world. There were no flowering plants, no grasses, no broad-leaved canopy trees as they exist today. Instead, the landscape was dominated by lycopsids, tree-sized relatives of modern club mosses, alongside corditalian conifers, seed ferns, and dense stands of horsetails, reaching heights of 10 meters or more. These plants formed continuous, moisture-retaining forests across equatorial Pangaea, their root systems interlocking through waterlogged soils that had accumulated organic matter across millions of years. Some of these forest systems extended without significant interruption across what are now multiple continents, functioning as single ecological units of continental scale. Within and beneath this vegetation, invertebrate life had reached its maximum Paleozoic diversity. Insects, benefiting from the oxygen-rich atmosphere and the absence of competing aerial predators, achieved body sizes that would be biologically impossible today. Meganeuropsis permiana, a dragonfly refined from fossil evidence in what is now North America, possessed a wingspan approaching 70 centimeters, larger than many modern birds. Flight under those atmospheric conditions was energetically cheaper and mechanically more forgiving. Insects occupied niches across the full vertical range of the forest, from the soil surface to the upper canopy, processing plant material, consuming one another, and sustaining the food webs that larger animals depended on entirely. The wetlands and swamp margins of Permian Pangaea were among the most productive environments on land. Standing water covered vast, low-lying basins, particularly across regions corresponding to parts of modern Europe, China, and North America, where river systems draining the interior highlands deposited sediment into broad, shallow floodplains. These environments supported an extraordinary range of amphibians, animals that, unlike their small modern descendants, had not yet been displaced from ecological dominance by fully terrestrial reptiles. Temnospondyls, the most diverse of these groups, ranged in body length from less than a meter to more than 3 meters in some genera. They occupied roles across the food chain from mid-level predator to apex hunter. Their anatomy was broadly crocodilian in functional terms. Low-slung, laterally, flattened, adapted for ambush at the water's edge. But their evolutionary origins were entirely distinct. They represented a lineage that had been developing independently for more than 60 million years by the late Permian. The terrestrial ecosystems of this period were governed by a fauna that, viewed from the perspective of modern biology, appears almost deliberately alien. The dominant large animals were not reptiles in the conventional sense, nor mammals, but a group called therapsids, sometimes referred to as mammal-like reptiles, though the term is a simplification. Therapsids were the evolutionary stage from which mammals would eventually descend. In the late Permian, they had diversified into a range of forms that occupied virtually every large-bodied ecological role available on land. Herbivores such as Dicynodon and its relatives, many of them stocky, barrel-bodied animals with keratinous beaks adapted for cropping vegetation, existed in enormous numbers. Their fossil records are distributed across multiple continents, suggesting population densities comparable to large modern ungulates. Some Dicynodont genera are estimated to have ranged across territories spanning thousands of kilometers, following seasonal resource gradients across the interior of Pangaea. Alongside the herbivores, and dependent on them, were the carnivores, Gorgonopsians, large, active therapsids with elongated canine teeth in some species exceeding 15 centimeters in length, were the dominant apex predators across much of the late Permian landscape. Their skeletal anatomy indicates a posture and gait significantly more erect than that of earlier reptilian predators, suggesting a degree of agility consistent with active pursuit hunting rather than simple ambush. The largest Gorgonopsian and genera, including Inostransenvia from what is now Russia, reached body lengths of approximately 3.5 meters and masses estimated between 150 and 300 kilograms, broadly comparable in size and ecological function to large modern felids such as lions. They were, however, unrelated to lions in any close evolutionary sense. The reef systems fringing the shallow seas of the late Permian represented a parallel peak of biological complexity. Carbonate reefs constructed primarily by sponge and bryozoan colonies rather than the coral-dominated systems of later geological periods, extended along the margins of the Tethys Sea, a body of water separating the northern and southern sections of Pangaea along an equatorial belt. These reef structures had developed over tens of millions of years, accumulating framework material at rates of centimeters per century, and by the late Permian had achieved thicknesses and lateral extents comparable to modern reef systems. The biodiversity they supported was structured across multiple trophic levels with trilobites, arthropods that had persisted through several previous extinction events, still present as active components of shallow marine communities. Alongside trilobites were crinoids, brachiopods, foraminifera, and a range of early fish lineages. The open ocean, beyond the shallow reef margins, was populated by predatory fish, early shark relatives, and large marine reptiles beginning to occupy niches in the water column. Evidence from sedimentary deposits suggests that primary productivity in the surface waters of Panthalassa was substantial, supporting food webs of considerable complexity. Marine invertebrate diversity at the genus and species level was near its Paleozoic peak. By some estimates derived from fossil assemblage data, biodiversity was approaching its highest point in the entire Paleozoic era. The 300 million year span of animal life that preceded the Permian boundary. What is significant about this picture, taken in aggregate, is not simply that life was diverse, but that it was structurally integrated across multiple scale and environments simultaneously. Terrestrial and marine systems were not isolated from one another. Nutrient cycling, sediment transport, and atmospheric chemistry connected them through processes operating continuously over geological time. The forests stabilized soils and moderated local climates. The soils supported invertebrate communities that fed amphibians and small reptiles. Those animals supported larger predators. The reefs filtered and cycled marine nutrients. The oceans regulated atmospheric carbon and oxygen through biological drawdown and sequestration. Each component of the system was embedded in and dependent upon the others. These relationships had taken hundreds of millions of years to establish. Evidence drawn from paleosol analysis, isotopic records in carbonate rock, and the stratigraphic distribution of fossil assemblages indicates that in the late Permian, roughly 252 to 260 million years ago, All of these systems were operating within stable, long-established parameters. Climate models reconstructed from proxy data suggest global mean temperatures in the range of 20 to 22 degrees Celsius, warmer than today, but within bounds that life had adapted to over many millions of years. Sea levels were relatively stable. Atmospheric carbon dioxide concentrations, while higher than pre-industrial modern values, were consistent with the biological productivity observed in the fossil record. By every measurable indicator available to modern science, this was a world at a peak, not moving toward collapse, not exhibiting early warning signals detectable in the geological record at this distance, but sustaining a biological complexity that had required the entirety of the Paleozoic era to construct. The system was not fragile in any conventional sense. It had survived glaciations, sea level fluctuations, and earlier extinction pulses. It had recovered from disruptions and rebuilt biodiversity across timescales of millions of years on multiple previous occasions. That resilience, built through deep time and expressed across every ocean basin and continent, would shortly prove insufficient. Not because the system was weak, but because what was coming had no precedent in the history of life on Earth. Among the most consequential evolutionary developments of the Permian period was the emergence of a land fauna capable of sustaining complex, multi-layered predator-prey dynamics across large continental distances. The therapsids, the dominant vertebrate group on land during this interval, had by the Middle Permian diversified into an ecological structure of considerable sophistication. Their success was not accidental. It was the product of anatomical innovations accumulated across tens of millions of years, producing animals fundamentally better suited to active terrestrial life than the sprawling ectothermic reptiles that had preceded them. The most immediately striking of these animals, to any observer reasoning from the fossil record, were the Gorgonopsians, distributed across the southern landmass of Pangaea, corresponding to modern Africa and Russia in particular, where the most complete specimens have been recovered. Gorgonopsians represented the culmination of a predatory lineage. That lineage had been refining its basic body plan for roughly 30 million years by the late Permian. Their skulls were elongated and heavily built, with temporal fenestrae, openings in the skull behind the eye socket that allowed for the attachment of significantly enlarged jaw musculature. In the largest species, the temporal region of the skull accounts for nearly half its total length, a proportion that indicates bite forces and gape widths well in excess of those produced by earlier Permian carnivores. The canine teeth of Gorgonopsians are among the most consistently documented features in the fossil record of this group. In Inostrancevia alexandri, the largest known species, upper canines reached lengths of approximately 15 centimeters, measured along the outer curve. These teeth possessed a lateral compression and fine serration along the posterior edge, consistent with tissue-cutting function rather than simple puncture. This morphology is functionally analogous, though not homologous, to the saber-tooth condition developed independently in several later mammalian lineages. It implies a similar prey acquisition strategy: rapid incapacitation of large-bodied prey through deep wounds to the neck or flank rather than sustained biting pressure. The prey available to these animals was, by late Permian standards, abundant dicynodonts, the most species-rich and numerically dominant herbivores of the period. Grazed and browsed across the floodplains, river margins, and open woodland environments of Pangaea. Their populations left behind some of the densest accumulations of vertebrate fossils known from the entire Paleozoic. Dicynodon itself, a genus represented by numerous species across what are now multiple continents, was a medium to large herbivore. Body lengths ranged from 1 to 2 meters, with masses estimated between 50 and several hundred kilograms depending on species. Their dentition had, in most lineages, been almost entirely replaced by a keratinous beak. Only two prominent tusk-like canines were retained in many genera. This beak, analogous in function to that of a large tortoise or a modern parrot, was capable of processing tough fibrous vegetation. The adaptation was well suited to the seed ferns and woody ground cover that formed the lower layer of Permian terrestrial ecosystems. Dicynodonts appear, from fossil distribution data, to have been highly mobile animals. Their remains have been recovered from sedimentary sequences deposited across a latitudinal range spanning from equatorial regions to high southern latitudes, suggesting either broad ecological tolerance or seasonal migration patterns comparable in scale to those of large modern ungulates. The implication, supported by the numerical abundance of their fossils relative to those of contemporary predators, is that dicynodont herds constituted a prey base of substantial and reliable biomass. They were the foundational resource upon which the entire large predator guild of the late Permian was built. Between the apex gorgonopsians and the herbivorous dicynodonts, the Permian terrestrial ecosystem included a range of intermediate predators filling niches across body size and hunting strategy. Therocephalians, another therapsid group, more closely related to the mammalian lineage than the gorgonopsians, occupied a broad range of body sizes, from small insectivores to mid-sized carnivores capable of taking prey comparable to their own mass. Some therocephalian genera show skeletal features, including details of the palate and nasal region, that have been interpreted as consistent with at least partial endothermy, internal heat generation. This adaptation would have extended their activity into temperature ranges inaccessible to fully ectothermic competitors. If this interpretation is correct, it represents an early expression of the physiological trajectory that would eventually produce fully warm-blooded mammals. The process remained incomplete and variable across the group during the Permian. Cynodontia, a third therapsid lineage, was present in the late Permian but not yet ecologically dominant. These animals, small to medium in body size, with increasingly mammal-like skull and dental anatomy, occupied the lower and middle levels of the terrestrial food web, feeding on insects, small vertebrates, and possibly plant material, depending on species. Their significance in the late Permian ecosystem was modest relative to their later importance, but their anatomical trajectory was already moving toward the body plan that would eventually characterize the first true mammals approximately 20 million years later. Alongside the therapsids, the Permian landscape was shared by more archaic reptilian lineages that had persisted from earlier periods. Parasaurs, large, heavily built herbivores with broad, flattened skulls and bodies armored with osteoderms embedded in the skin, reached impressive sizes. They attained body lengths of up to 3 meters and masses approaching 1 metric ton in the largest genera. They were not therapsids, but rather members of a separate reptilian lineage, and their bulk and armor likely functioned as passive defense against gorgonopsian predation. Fossil evidence from sites in Russia and South Africa places parasaurs and large gorgonopsians in the same depositional environments, consistent with a direct predator-prey relationships sustained across millions of years. The amphibians of this period, though declining relative to their dominance in earlier Carboniferous ecosystems, retained a significant presence in riparian and wetland habitats. Large temnospondyls occupied the margins of rivers and lakes across equatorial Pangaea. Some genera exceeded 2 meters in body length and functioned as ambush predators of fish, smaller tetrapods, and any terrestrial animal that approached the water's edge within strike range. Their role in the ecosystem was broadly analogous to that of modern large crocodilians, though their evolutionary relationship to those animals is entirely separate. In environments where permanent water was available, temnospondyls were consistent components of the vertebrate fauna. Their populations appear to have been stable and, geographically widespread throughout the late Permian. What emerges from the combined fossil record of Permian terrestrial vertebrates is a picture of an ecosystem operating through well-established, mutually reinforcing relationships. These relationships connected producers, consumers, and predators in a structure built across geological time and expressed through anatomical specialization at every trophic level. The hunting behavior of Gorgonopsians was calibrated, in an evolutionary sense, to the defensive and locomotory capabilities of Dicynodonts. The body size of Parasaurs was in part a response to the predatory pressure of the largest carnivores. The physiological advances appearing in Therocephalians and Cynodonts reflect competitive pressures operating across millions of generations in ecosystems of genuine ecological complexity. This was not a primitive or unstable biological world. It was a mature one, the product of an evolutionary process spanning the entirety of the Paleozoic. Refined through successive episodes of competition, adaptation, and ecological reorganization, the animals moving across the floodplains and through the forests of late Permian Pangaea were not transitional or experimental in any simple sense. They were precisely fitted to the environments they inhabited, participants in food webs of measurable stability. They were representatives of lineages that had already survived considerable geological and climatic change. That stability and the specialization it had produced would become a liability when the conditions sustaining it began, irreversibly, to change. While the terrestrial ecosystems of late Permian Pangaea supported their complex fauna of therapsids and giant amphibians, the oceans covering the majority of the planet's surface sustained a biological world of parallel, and in some respects greater, complexity. Panthalassa, the vast global ocean surrounding the supercontinent, and the Tethys Sea cutting eastward through Pangaea's equatorial margin, together represented the largest continuous marine habitat in Earth's history to that point. The shallow water environments fringing the continental margins were, by the late Permian, among the most structurally complex marine habitats the planet had yet produced. Their combined surface area exceeded that of all modern oceans, and the biological communities they supported had been developing, largely without catastrophic interruption, across the full span of the Paleozoic era. More than 300 million years of continuous marine evolution. Reef systems constructed primarily by calcareous sponges, specifically the hypercalcified sponges known as Archaeocyath descendants, and, more dominantly, the Sphinctozoan and Inozoan sponge groups, extended along the margins of the Tethys Sea for thousands of kilometers. These structures were not coral reefs in the modern sense. The scleractinian corals that would eventually dominate Mesozoic and Cenozoic reef systems had not yet diversified into ecological prominence. Instead, the framework-building role was filled by sponge colonies, bryozoans, and encrusting organisms that accumulated carbonate material at rates consistent with the warm, shallow, well-oxygenated conditions of the Tethyan margin. Within and around these reef structures, invertebrate diversity reached its Paleozoic maximum. Brachiopods, filter-feeding shellfish that superficially resemble bivalves but are anatomically entirely distinct, dominated the seafloor communities of shallow Permian seas. They achieved a level of ecological dominance they would never again reach after the period's end. Thousands of species have been described from Permian marine deposits, representing a diversity of form, size, and ecological function. This diversity spanned from encrusting reef-associated forms to free-living animals on open soft sediment. Their numerical abundance in some Permian limestone sequences is sufficient to constitute the majority of the rock's volume. That abundance represents a measure of population density, with few equivalents in the fossil record of any other animal group. Trilobites, the iconic Paleozoic arthropods that had first appeared more than 500 million years earlier, were still present in late Permian marine communities. Their diversity, however, had been substantially reduced by a series of earlier extinction events. The species that survived into the final stages of the Permian were predominantly small and occupied restricted ecological niches. They were largely confined to specific substrate types and depth ranges. Their persistence across such an enormous span of geological time is a measure of their fundamental biological resilience. By the late Permian, they represented a lineage in long-term decline, rather than active diversification. Fossil assemblages from Permian marine sequences show trilobite diversity continuing to contract through the period. The remaining diversity became concentrated into progressively fewer genera as the epoch advanced. The water column above the seafloor was populated by a range of active predators operating across multiple depth ranges. Nautiloid cephalopods, relatives of the modern nautilus, though representing numerous distinct lineages, were present as mid-water predators, using jet propulsion and a chambered shell for buoyancy regulation to pursue prey across depth gradients. Some Permian nautiloid genera reached shell diameters exceeding 30 centimeters, positioning them as significant predators within the pelagic food web. Ammonoids, a related but more rapidly evolving group of cephalopods, had also diversified considerably. After recovering from near extinction during an earlier Permian crisis event, ammonoids occupied a range of ecological roles, from slow-moving benthic scavengers to faster pelagic hunters. Fish diversity in Permian oceans was substantial and structurally important to marine food webs at multiple levels. Actinopterygian ray-finned fish, the lineage that would eventually produce the vast majority of modern fish species, were already present and diversifying. They had not yet achieved the ecological dominance they would hold in later geological periods. Alongside them, chondrichthyans cartilaginous fish, including the ancestors of modern sharks and rays, occupied apex and mid-level predatory roles. These predators hunted in both shallow and open water environments. Some Permian shark lineages produced body lengths estimated from tooth and scale fossil evidence at 3 meters or more. Such sizes were sufficient to place them among the largest active predators in the marine ecosystem. The deepest and most open waters of Panthalassa are, by the nature of deep-sea preservation, less thoroughly documented in the fossil record than shallow water environments. Sediment accumulation in deep ocean basins is slow, and the geological processes that return ancient deep-sea deposits to the surface through subduction and abduction at tectonic plate margins destroy or alter most biological material preserved within them. What survives in the form of radiolarian cherts and pelagic limestone sequences indicates that primary productivity in the open ocean surface waters was sustained by diverse communities of planktonic organisms. These included radiolarians, foraminifera, and various algal groups, whose organic material fueled food webs extending from microscopic filter feeders to large active predators. The geochemical record, preserved in late Permian marine sediment, provides quantitative evidence for the stability of these ocean systems in the period immediately preceding the extinction event. Oxygen isotope ratios in carbonate shells indicate sea surface temperatures in the Tethys region of approximately 25 to 28 degrees Celsius, warm but within the range that reef building and high-diversity shallow marine communities had long occupied. Carbon isotope values in the same sedimentary sequences show relatively stable patterns of organic carbon burial and marine productivity, without the sharp excursions that characterize periods of severe ecological disruption. Sulfur isotope records indicate iron chemistry within normal Paleozoic parameters, with no evidence of widespread anoxia. Oxygen depletion in shallow water environments during this interval. This geochemical stability is significant because it establishes at baseline the late Permian Ocean, as recorded in the sedimentary and isotopic evidence, was not a system under acute stress in the interval before the extinction horizon. It was a system in equilibrium, chemically buffered, biologically productive, structured through ecological relationships that had been established and reinforced across hundreds of millions of years of continuous marine evolution. The reef systems were growing. The invertebrate communities were diverse. The predator-prey dynamics of the water column were functioning through well-integrated food webs, connecting primary producers at the surface to consumers at every depth. Foraminifera, single-celled organisms that secreted intricate calcium carbonate shells and lived in enormous numbers throughout the water column and on the sea floor, provide one of the most precise records of late Permian marine vintage available to modern science. Their shells, preserved in vast quantities in sperm-earth limestone sequences across Iran, China, and the Tethyan margin generally, record changes in water temperature, chemistry, and productivity at a resolution approaching individual centuries in some sequences. In the late Permian interval before the extinction horizon, these records show a marine system of high diversity and apparent stability. Species accumulations in individual limestone beds sometimes exceeded several hundred foraminiferan taxa per sample. This density of biological complexity reflects not just diversity, but the ecological depth and interconnection of the system producing it. The oceans of the late Permian were, in the most precise terms available from the geological record, a mature biological system, one that had been constructed across the entirety of the Paleozoic through the cumulative action of evolution, ecological interaction, and geochemical cycling. Their complexity was not superficial. It was structural, expressed at every level from ocean chemistry to apex predation, and maintained through feedback mechanisms operating simultaneously across biological and geochemical scales. What that complexity represented, ultimately, was not permanence. It was depth, and depth, when disruption arrives faster than recovery is possible, becomes a measure of what stands to be lost. The terrestrial plant communities of the late Permian period represented the most expansive and structurally complex vegetation systems the planet had yet assembled. Across the equatorial and mid-latitude zones of Pangaea, continuous forest-covered landmasses of continental scale, producing ecosystems of layered biological complexity that had been developing without fundamental interruption. Since the Carboniferous period some 50 million years earlier, these were not forests recognizable by modern standards. Their composition, architecture, and the physical conditions sustaining them were products of a world operating under atmospheric and climatic parameters substantially different from those of the present, and the organisms constituting them belonged almost entirely to lineages that no longer exist. The dominant plant groups of Permian forests were gymnosperms, seed-producing plants that reproduced without flowers or fruit. Glossopterids, a group of seed ferns characterized by distinctive tongue-shaped leaves, formed extensive forests across the high-latitude southern regions of Pangaea. The landmass corresponding to modern Antarctica, Australia, India, South America, and Southern Africa was then joined into a single continuous territory. Glossopterid forests grew under conditions of strong seasonal light variation at high latitudes. Their growth rings, preserved in fossilized wood specimens recovered from Antarctica and Australia, indicate periods of rapid summer growth followed by winter dormancy. A physiological strategy broadly analogous to that of modern boreal forests, though produced by entirely unrelated plant lineages. In the equatorial and lower latitude zones, the forest composition shifted toward caudatalian conifers, tall, straight-trunked trees with strap-like leaves, reaching heights estimated from trunk diameter fossils at 20 to 30 meters. These forests also contained tree-sized seed ferns, horsetails, and persistent lycopsid stands in waterlogged lowland settings. The forests were structurally stratified in the vertical dimension, with a high canopy of corditalians and seed ferns above a mid-layer of smaller woody plants. A ground layer of ferns, mosses, and herbaceous-like copsids occupied the forest floor. This vertical stratification created a range of distinct microhabitats within a single forest stand, each supporting its own community of invertebrates, small vertebrates, and decomposers. The soils beneath these forests were the product of millions of years of biological activity. Root penetration, organic matter accumulation, and the biochemical activity of fungal networks had produced deep, structured soil profiles capable of retaining moisture, cycling nutrients, and supporting plant communities through seasonal variation. Paleosol sequences ancient soil horizons preserved in the sedimentary record. From Permian deposits across multiple continents, document soil depths and organic content consistent with stable, mature terrestrial ecosystems operating under sustained vegetation cover. These soils were not simply a substrate for plant growth. They were active biological systems in their own right, processing organic material at rates that regulated atmospheric carbon and supported the invertebrate communities upon which larger animals ultimately fed. The moisture dynamics of Permian forests were governed in part by the forests themselves. Dense vegetation cover reduces surface albedo, increases evapotranspiration, and generates local precipitation recycling. Through these processes, forests effectively maintain the humid conditions that sustain them. In the broad lowland basins of equatorial Pangaea, where topography was relatively flat and drainage was slow, this feedback produced persistently waterlogged environments supporting dense swamp forests with high standing biomass. Fossilized peat deposits from these environments, now preserved as coal seams in Permian-age rock sequences across China, Europe, and North America, record organic accumulation rates consistent with highly productive, continuously vegetated wetland systems. These systems were maintained across timescales of hundreds of thousands of years. The insect communities inhabiting these forests were, in terms of diversity and ecological function, among the most complex terrestrial invertebrate assemblages in the fossil record of the Paleozoic. Oxygen concentrations near or above 30% by volume during parts of the Permian supported the respiratory physiology of large-bodied insects through passive diffusion, a mechanism that imposes strict size limits under modern atmospheric conditions. At higher oxygen partial pressures, however, much larger body sizes became viable. Fossil evidence from Permian deposits documents not only the giant odonate relatives discussed previously, but a broad range of large-bodied forms across multiple insect orders. These included cockroach relatives, early beetle lineages, and diverse hemipteran groups. These insects occupied decomposer, herbivore, and predator roles throughout the forest ecosystem, processing plant material and consuming one another. They also constituted the primary food source for insectivorous vertebrates across a range of body sizes. The vertebrate fauna associated with Permian forest and woodland environments extended beyond the large therapsids of the open floodplains. Small to medium-sized insectivorous and omnivorous tetrapods, including early cynodont therapsids, small therocephalians, and various reptilian lineages, occupied the forest interior, exploiting the invertebrate productivity of the forest floor and understory. Their presence in the fossil record of forested environments is less complete than that of larger animals from open depositional settings, partly because forest soils are generally poor preservation environments. Trackway evidence and isolated skeletal material from several Permian localities nevertheless indicate a diverse small vertebrate fauna intimately connected to the forest ecosystem. Amphibians retained a consistent presence in the riparian and swamp forest environments. Permanent or semi-permanent water availability met their reproductive requirements. The interface between aquatic and terrestrial habitats in Permian forested wetlands was a zone of particularly high biological activity. This transitional environment concentrated nutrient inputs from both systems, supporting dense populations of invertebrates, fish, and small tetrapods that in turn sustained larger predators, documented through fossil assemblages from Permian coal swamp deposits. This ecological interface functioned as a biological connectivity zone, linking the productivity of aquatic systems to the food webs of the surrounding forest. The geographic extent of Permian forest cover across Pangaea was not uniform. Interior regions of the supercontinent, distant from oceanic moisture sources, experienced significantly drier conditions. This interpretation is supported by paleosol evidence showing reduced organic content and calcrete formation consistent with seasonal aridity. In these interior zones, forest gave way to open woodland, shrubland, and in the most arid regions, near desert conditions with sparse vegetation. This continent-scale moisture gradient produced a mosaic of vegetation types rather than a single uniform forest. The animal communities of drier interior regions, documented from redbed sedimentary sequences in Russia, China, and southern Africa, were distinct in composition. They differed from those of the humid equatorial forests, though connected through the movement of wide-ranging species across the intervening gradient. What the combined evidence of paleobotany, paleosol analysis, coal geochemistry, and fossil vertebrate assemblages describes is a terrestrial biological system of genuine structural depth. The forests of the late Permian were not simply collections of plants. They were integrated ecological systems in which vegetation, soil biology, atmospheric chemistry, hydrology, and animal communities functioned as interdependent components. Each component sustained and was sustained by the others through processes operating simultaneously across spatial scales from the individual root system to the continental watershed. The stability of these systems, as recorded in the geological evidence, was real and measurable. Forest composition remained broadly consistent across millions of years of Permian time, as indicated by the stratigraphic continuity of plant fossil assemblages. This continuity is visible in Permian age sedimentary sequences. Soil profiles show sustained biological activity, without evidence of repeated disruption and recovery. Coal deposits, accumulating continuously across hundreds of thousands of years, attest to uninterrupted wetland productivity maintained through multiple glacial and interglacial cycles of the early Permian and into the period's warmer later stages. This continuity was the product of deep time, of biological and ecological processes allowed to operate and reinforce one another across spans of time measured in tens of millions of years. The forests of the late Permian had not simply grown. They had matured into systems of self-sustaining complexity, resilient across the full range of environmental variation the Permian world had presented them with up to that point. That resilience had limits, and those limits were approaching. The geological record of the late Permian does not transition abruptly from stability to catastrophe. Between the peak of biological complexity documented in the fossil and geochemical evidence and the acute phase of the extinction event itself, there exists an interval, spanning several million years, and in some regions considerably less, during which measurable changes in climate, ecology, and environmental chemistry began accumulating. These changes were not, in their early stages, of a magnitude sufficient to produce immediate or widespread biological collapse. They were, however, directional, and in a biological world as structurally integrated as that of the late Permian, directional environmental style change sustained across times long enough to exceed the adaptive capacity of established East Coast ecosystems, carries in consequences that compound progressively rather than remaining contained. The first signals detectable in the geological record are climatic, paleosol sequences from Permian continental depositional basins, including sequences in the Karoo Basin of southern Africa, one of the most thoroughly studied late Permian terrestrial archives available, show a transition toward drier conditions beginning in the interval several million years before the extinction horizon. The specific indicators are consistent across multiple independent proxies, increasing calcrete formation in soil profiles, indicating reduced moisture availability, decreasing organic carbon content in paleosols, reflecting reduced plant productivity and organic matter input, and shifts in the isotopic composition of pedogenic carbonates, consistent with warming and increased evaporation. These signals do not appear simultaneously or uniformly across all regions, but their occurrence across multiple geographically separated sequences suggests a global rather than locally driven process. Pollen and spore assemblages preserved in late Permian sedimentary sequences provide a complementary line of evidence. In several regions, including deposits from China, Russia, and the Karoo Basin, the taxonomic composition of pollen assemblages shifts in the interval preceding the extinction horizon. The relatives of moisture-dependent plant taxa declines, while the representation of drought-tolerant or opportunistic forms increases. These shifts are gradual in some sequences and more abrupt in others, reflecting regional variation in the timing and intensity of climatic stress. In the most detailed Chinese sequences, where sediment accumulation rates are sufficient to resolve changes at a resolution approaching tens of thousands of years, the pollen record shows episodic fluctuations in plant community composition, superimposed on a longer-term trend toward reduced diversity and increased representation of stress-tolerant taxa. The carbon isotope record of late Permian marine carbonates shows perturbations reaching the main extinction horizon that have been interpreted in current geochemical models as reflecting instability in the global carbon cycle. Carbon isotope values in carbonate sequences from the Tethys margin show negative excursions, reductions in the ratio of carbon-13 to carbon-12 that indicate episodic inputs of isotopically light carbon into the ocean atmosphere system. The sources of such inputs in the geological context of the late Permian could include volcanic outgassing, oxidation of organic-rich sediments, or destabilization of methane hydrates on continental shelves. The excursions, visible in pre-extinction Permian sequences, are smaller in magnitude than those associated with the extinction horizon itself, but their presence indicates that the carbon cycle was subject to periodic disruption before the catastrophic phase began. Marine invertebrate assemblages in the late Permian show evidence of ecological reorganization in some regions during the interval preceding the extinction. Brachiopod diversity in certain Tethyan sequences begins declining measurably before the extinction horizon. Taxonomic richness reduces and assemblage composition shifts toward forms tolerant of reduced oxygen or increased temperature. Reef communities in some localities show evidence of stress, reduced calcification rates and increased prevalence of microbial mat communities relative to metazone framework builders. These changes are consistent with deteriorating water chemistry or temperature conditions. The signals are not universal across all late Permian marine sequences, and their interpretation remains subject to ongoing scientific discussion. Their occurrence in multiple independent sections nevertheless suggests that marine ecosystems were experiencing measurable stress before the main extinction pulse. Terrestrial vertebrate assemblages in the Karoo Basin, where the stratigraphic record of late Permian land vertebrates is among the most complete available globally, show compositional changes in the interval preceding the extinction horizon. Detailed analysis of fossil vertebrate assemblage zones in the Karoo sequence indicates a reduction in the diversity of dicynodont species in the latest Permian interval, with some genera disappearing from the record before the extinction horizon. The fauna shows a progressive simplification toward lower species richness and reduced ecological complexity. Whether these changes reflect genuine pre-extinction biodiversity loss or are partly an artifact of preservational variation remains a subject of active scientific investigation. The pattern across multiple independently analyzed Karoo sections is sufficiently consistent to suggest a real biological signal. The physical environment of the late Permian was also shifting in ways detectable through sedimentological evidence. River systems in some continental basins show changes in depositional style in the interval approaching the extinction. Transitions from meandering river systems, which require stable vegetation cover to maintain channel banks, toward braided river systems. Braided systems are characteristic of reduced vegetation cover and increased sediment supply. This transition, documented in Permian-age fluvial sequences from several continents, implies a reduction in the density and stability of terrestrial plant communities. The reduction was sufficient to alter the hydrological behavior of major drainage systems. The vegetation that had stabilized soils and regulated moisture across Permian landscapes was, in at least some regions, beginning to thin and contract. Taken individually, each of these signals is interpretable within the range of normal geological and ecological variability. Climatic shifts, localized faunal turnovers, and carbon cycle perturbations are recurring features of the geological record. Such changes do not necessarily indicate approaching catastrophe. What distinguishes the late Permian pre-extinction interval is the convergence of multiple independent proxies: climatic, geochemical, paleoecological, and sedimentological, all indicating change in the same direction across the same temporal interval. The signals are not identical in timing or magnitude across all regions, reflecting the spatial complexity of a global system responding to forcing that was itself geographically variable. Their collective pattern, assembled from deposits distributed across multiple continents and ocean base, describes a planetary system moving systematically out of the equilibrium state that had sustained Permian biodiversity. The ecosystems of the late Permian were not static. They had always existed within a range of environmental variation, and the biological communities constituting them had adaptive capacity, built through millions of years of exposure to fluctuating conditions. But adaptive capacity operates within limits defined by the rate and magnitude of environmental change. When environmental forcing exceeds those limits, when change arrives faster than evolutionary and ecological adjustment can track, the integrated structure of an ecosystem does not simply bend. It begins, progressively and then acceleratingly, to break. The evidence preserved in the geological record of the late Permian describes exactly this process in its earliest stages. A world still recognizably intact, still functioning through the ecological relationships built across the Terregostoic. But in the chemistry of its soil and oceans, and in the composition of its biological communities, the first measurable consequences of forces that had not yet reached their full expression. Those forces were building beneath the surface of the planet, in a region corresponding to what is now northern Russia. A geological process of extraordinary scale was approaching the threshold at which its effects would become impossible for the living world to absorb. Beneath the surface of the late Permian Earth, geological processes had been accumulating energy across timescales measured in millions of years. The mechanism responsible for what followed was not a single event, nor a process initiated by any surface-level environmental condition. It originated in the deep mantle, in the slow thermal convection of partially molten rock, operating at depths of several hundred kilometers below the crust, where temperatures exceed 100 degrees Celsius and pressure renders material behavior fundamentally different from anything observable at the surface. Within this deep system, a thermal anomaly, a mantle plume, in the terminology of modern geodynamics, had been rising toward the base of the lithosphere for an extended period. When it arrived, the consequences would be recorded in the geological record of every continent and every ocean basin on the planet. The surface expression of this process was the Siberian Traps, a large igneous province of a scale rarely matched in the geological record of the past 500 million years. The term trap derives from a Swedish word for staircase, referring to the stepped topography produced by successive lava flows of varying resistance to erosion. The Siberian Traps, as they are preserved today in the sedimentary and volcanic sequences of central and northern Russia, cover an area of approximately 2 million square kilometers, roughly equivalent to the combined area of Western Europe. Current estimates, derived from the volume of preserved volcanic material and reconstructions accounting for erosional loss, suggest that the total volume of magma erupted during the main phase of Siberian Traps volcanism was on the order of 2 to 3 million cubic kilometers. This figure represents one of the largest volcanic outpourings in the geological record of the Phanerozoic Eon. The initiation of major eruption activity in the Siberian Traps is dated through high-precision radiometric methods including uranium lead zircon geochronology to approximately 252 million years ago. That date coincides, within current analytical resolution, with the onset of the end Permian extinction event. The precision of modern radiometric dating has progressively reduced the uncertainty around this correlation. Current published age constraints place the onset of major volcanism and the beginning of the extinction horizon within a temporal overlap of less than 300,000 years, and potentially considerably less. This temporal coincidence, replicated across multiple independently dated sequences from Russia, China, and other regions, constitutes the primary evidence for a causal relationship between the Siberian Trap eruptions and the extinction event. The physical character of the eruptions was not that of the explosive, ash-dominated volcanic events most familiar from the human historical record. The Siberian Traps were predominantly effusive, meaning that magma reached the surface not through explosive fragmentation, but through sustained outpouring of fluid basaltic lava across broad fissure systems. Individual lava flows, identifiable in the preserved volcanic stratigraphy of the Siberian sequence, reached thicknesses of tens of meters, and the accumulated stack of flows in the central portions of the province exceeds 3 kilometers in total thickness. The lateral extent of individual flows, reconstructed from correlations between geographically separated sections, indicates that single eruptive episodes produced lava fields extending hundreds of kilometers from their source vents. These flows inundated landscapes at rates that would have made terrestrial survival in the affected regions effectively impossible. The rate of eruption, rather than the total volume alone, is critical to understanding the environmental consequences. Current geochemical and stratigraphic models suggest that the most intense phase of Siberian Traps volcanism, responsible for the majority of the total erupted volume, was concentrated within an interval of approximately 1 million years or less. Some models indicate that a significant fraction of the total magma output was emplaced within a few 100,000 years. This rate of magma production implies volcanic gas emission rates far exceeding those of any eruption in the human historical record. These emissions were sustained continuously across timescales orders of magnitude longer than any historically observed volcanic episode. The gases released by basaltic volcanism at this scale constitute the primary mechanism through which the Siberian Traps affected global environmental systems. Carbon dioxide, released directly from the magma as it depressurized at the surface, was emitted in quantities estimated by current geochemical models at between 10,000 and 40,000 gigatons across the main eruptive phase. This range reflects genuine uncertainty in the volatile content of the source magma and the proportion of gas retained versus released. Sulfur dioxide emissions, which produce rapid but shorter duration atmospheric cooling through the formation of stratospheric sulfate aerosols, were also substantial. Estimates from sulfur isotope and fluid inclusion data suggest repeated large magnitude sulfur pulses associated with individual eruptive episodes. Halogens, including chlorine and fluorine, were released in quantities sufficient to have produced significant stratospheric ozone depletion. According to current atmospheric chemistry models, this ozone depletion increased ultraviolet radiation reaching the surface, a factor that substantially amplifies the environmental impact of the Siberian traps. Beyond the direct volcanic emissions is the geological setting into which the magma was intruded. Beneath the region of central Siberia affected by the plume, thick sequences of sedimentary rock had accumulated over hundreds of millions of years, including extensive coal deposits, evaporite sequences containing large reservoirs of sulfate and halide minerals, and organic-rich shales. As the rising magma intruded into these sedimentary sequences, forming sills and dikes that penetrated the country rock rather than reaching the surface directly, it thermally decomposed the surrounding material. This process, known as contact metamorphism, converts coal into coke and releases the organic carbon stored within it as carbon dioxide and methane. It drives chemical reactions within evaporite sequences that release sulfur dioxide, methane, and halocarbons directly into the subsurface. From there, these gases migrate upward through faults and fractures to the atmosphere. Evidence for this process comes from multiple independent sources. Carbon isotope anomalies in Permian Triassic boundary sequences worldwide show signatures consistent with the addition of isotopically light carbon, the geochemical fingerprint of thermally decomposed organic material. Rather than purely mantle-derived volcanic carbon, nickel anomalies in boundary sediments, reflecting the geochemical signature of the Siberian magmas, have been identified in sequences from China, Italy, and the Arctic, consistent with global atmospheric transport of volcanic material. Breccia, pipes, geological structures formed by the explosive release of gas through overlying rock, have been identified within the Siberian Traps province in large numbers. Their presence indicates that subsurface gas generation and pressurization was sufficient to produce violent degassing events capable of injecting material directly into the stratosphere. The combined effect of direct volcanic emissions and contact metamorphic gas generation produced an atmospheric forcing of a magnitude and rate that the planetary system had not experienced within the preceding tens of millions of years. The carbon dioxide released across the main eruptive phase represents a perturbation to the atmospheric carbon budget orders of magnitude larger than current anthropogenic emissions. It was delivered not instantaneously, but across a sustained interval that prevented the normal geochemical buffering mechanisms, silicate weathering, organic carbon burial, ocean carbonate dissolution, from compensating at a rate sufficient to maintain equilibrium. The planet's climate system, its ocean chemistry, and ultimately its biological communities were now subject to forcing on a scale, and at a rate for which the geological record of the Paleozoic offered no precedent. The eruptions had begun, and the world that had taken 300 million years to assemble was about to begin coming apart. The gases released by the Siberian Traps volcanism did not remain confined to the immediate vicinity of their source. Within timescales governed by atmospheric circulation, days to weeks for lower atmospheric mixing, months to years for stratospheric distribution, the chemical products of the eruptions dispersed across the full extent of the planetary atmosphere. What followed was not a single discrete event, but a sustained, compounding alteration of the atmospheric composition that the living world had occupied without fundamental disruption across the entirety of the Paleozoic. The atmosphere of the late Permian was not simply polluted in any local or recoverable sense. It was chemically restructured at a global scale and across a time frame that exceeded the capacity of biological systems to absorb. Carbon dioxide constitutes the most consequential of the Siberian TRAPSIST emissions in terms of long-duration environmental impact. As a greenhouse gas, carbon dioxide traps outgoing infrared radiation from the Earth's surface, reducing the rate at which the planet loses heat to space and producing a corresponding increase in surface temperature. The relationship between atmospheric carbon dioxide concentration and surface temperature is nonlinear and influenced by numerous feedback mechanisms, but the fundamental physical principle is well established across multiple lines of evidence, including ice core records, paleoclimate proxy data, and radiative transfer calculations. This increase produced a radiative forcing equivalent to several watts per square meter, sustained across hundreds of thousands of years. The surface temperature response to this forcing, as reconstructed from oxygen isotope records in marine carbonates and phosphates from Permian Triassic sections, was substantial. Published estimates of end-Permian warming derived from multiple independent proxy systems converge on tropical sea surface temperature increases of between 8 and 15 degrees Celsius above late Permian baseline values across the boundary interval. The uncertainty range in these estimates reflects genuine variability in proxy preservation and calibration, but even the lower bound of this range represents a thermal perturbation of extraordinary biological significance. Modern physiological studies indicate that tropical marine organisms, already living near the upper thermal limits of their metabolic tolerance under ambient Permian temperatures, would have experienced conditions exceeding those limits across much of the low-latitude ocean. These conditions developed within the warming interval, reconstructed from the geological record. Sulfur dioxide, released in large quantities during individual eruptive pulses, produced atmospheric effects operating on shorter timescales than carbon dioxide, but with distinct and potentially severe biological consequences. Current geochemical models of the end-Permian carbon dioxide pulse, calibrated against carbon isotope records, and carbonate chemistry data from boundary sequences, indicate that atmospheric carbon dioxide concentrations may have doubled or more across the main eruptive phase. In the stratosphere, sulfur dioxide oxidizes to form sulfate aerosols, fine particles that reflect incoming solar radiation and produce rapid surface cooling on timescales of 1 to 3 years following major eruptive events. This cooling effect, documented in the historical record of large volcanic eruptions, including the 1991 Pinatubo eruption, which produced measurable global cooling of approximately 0.5 degrees Celsius, would have been replicated repeatedly and at substantially greater magnitude across the Siberian TRAPS eruptive sequence. The alternation between sulfur-driven cooling pulses and the sustained warming trend driven by carbon dioxide accumulation would have produced a pattern of rapid climate oscillation, superimposed on a longer-term warming trajectory. This combination of short-term instability and directional change would have been particularly disruptive to ecosystems adapted to stable climatic conditions. In the lower atmosphere, sulfur dioxide dissolves in atmospheric water vapor to form sulfuric acid, which precipitates as acid rain. The pH reduction associated with sulfuric acid deposition from large volcanic events is well documented in the geological record and in atmospheric chemistry models. During periods of intense Siberian TRAPS degassing, sulfur dioxide emissions sufficient to produce acid precipitation would have fallen across continental landscapes and ocean surfaces simultaneously. the precipitation may have approached pH values significantly below normal, potentially nearing 4 or lower in regions downwind of the eruption centers. The biological consequences of acid deposition at this scale operate through multiple mechanisms. These include direct tissue damage to plant foliage and gill surfaces of aquatic organisms, dissolution of calcium carbonate in soils, reducing the buffering capacity that normally moderates soil pH, and mobilization of aluminum and other metal ions into soil water and surface runoff. These processes produce toxic concentrations in freshwater systems. The halogen compounds released through contact metamorphism of evaporite sequences beneath the Siberian traps included methyl chloride, hydrogen chloride, and various chlorinated and brominated organic compounds. These compounds were generated by the thermal alteration of halide-bearing sedimentary rock and reached the stratosphere through the same explosive degassing pathways that delivered other volatile compounds to the upper atmosphere. In the stratosphere, halogen compounds participate in catalytic reaction cycles that destroy ozone molecules. Stratospheric ozone, concentrated in a layer between approximately 15 and 35 kilometers altitude, absorbs the majority of incoming solar ultraviolet radiation in the biologically damaging UVB wavelength range. This range extends from 280 to 315 nanometers. Depletion of the ozone layer increases the flux of UVB radiation, reaching the surface portion to the degree of ozone column reduction. This relationship is described by the Dobson unit framework and validated against observational data from modern ozone depletion events associated with anthropogenic halogen emissions. Evidence for elevated UV radiation during the end Permian event is preserved in the fossil record in the form of malformed spores and pollen grains recovered from Permian Triassic boundary sediments across multiple continental sections. Spore wall malformation, the production of abnormal, asymmetric, or multiply-fused spore structures, is a documented biological response to elevated UVB radiation, observable in modern plant populations exposed to increased ultraviolet flux. The abundance of malformed spores in boundary sediments from sections in Greenland, Canada, and China significantly exceeds background levels observed in pre-boundary Permian sequences, providing direct biological evidence for a UVB pulse coinciding with the extinction horizon. The magnitude of the malformation signal in some sections implies ozone column reductions sufficient to increase surface UVB flux by factors of 2 to 10 above pre-extinction baseline levels. Such increases would have placed severe physiological stress on surface-dwelling photosynthetic organisms and shallow water marine life simultaneously. Methane, released both by direct volcanic degassing and by the thermal decomposition of organic-rich sediments beneath Siberian traps intrusions, contributed to atmospheric warming. Methane is a significantly more potent greenhouse gas than carbon dioxide on a per-molecule basis over short timescales. Approximately 80 times more effective at trapping heat over a 20-year window, though it oxidizes to carbon dioxide in the atmosphere over timescales of approximately a decade. Large methane pulses would therefore have produced episodes of accelerated short-term warming superimposed on the sustained carbon dioxide-driven temperature increase, potentially driving surface temperatures briefly beyond the bounds reconstructed from the longer-term isotopic record. The cumulative atmospheric state produced by these simultaneous and interacting chemical perturbations was one for which no analog existed within the biological experience of late Permian organisms. The plants, animals, and microorganisms of the Permian world had evolved under atmospheric conditions that had varied across the Paleozoic but had never combined elevated carbon dioxide, reduced oxygen availability in some regions, acid precipitation, elevated UV radiation, and rapid temperature oscillations simultaneously and at global scale. Each of these stressors, applied individually and gradually, might have been survived through biological adaptation operating across many generations. Applied together, at rates exceeding those of evolutionary response, they constituted a compound perturbation with no biological precedent. The sky above the late Permian world was changing in ways that no organisms alive could perceive directly. There were no instruments to measure the shifting chemistry of the atmosphere, no means by which any living creature could recognize the accumulating toxicity of the air and rain. Survival was becoming progressively more difficult. The forests that had regulated moisture and stabilized soils for millions of years began to register the acid deposition in the chemistry of their tissues. The shallow marine organisms whose calcium carbonate shells recorded ocean chemistry with precision were beginning to experience water conditions outside the range their physiology had been built to tolerate. The atmosphere, which had sustained the complexity of the Permian world across hundreds of millions of years, had become something different, and the consequences of that transformation through the systems it touched. The oceans of the late Permian had sustained biological communities of extraordinary complexity across hundreds of millions of years of continuous marine evolution. Their chemistry, structured through the long-term interactions of biological productivity, atmospheric gas exchange, carbonate dissolution, and deep water circulation, had remained within parameters broadly compatible with diverse and abundant marine life throughout the Paleozoic. What the Siberian Traps volcanism initiated at the surface and in the atmosphere was, through a series of interconnected physical and chemical processes, translated into the marine environment with consequences that would ultimately prove more lethal to ocean life than any previous perturbation in the geological record of the Phanerozoic. The primary mechanism connecting atmospheric change to marine biological collapse was ocean warming. As atmospheric carbon dioxide concentrations rose across the eruptive interval, the transfer of heat between the atmosphere and the ocean surface drove progressive warming of the upper ocean. This process is governed by the temperature differential between the two systems and by the solubility of gases in seawater. Sea surface temperatures in the tropical Tethys, as reconstructed from oxygen isotope and clumped isotope data in carbonate fossils from boundary sections, rose substantially. The increase was consistent with atmospheric forcing estimates, reaching test levels that, in the low-latitude shallow water environments, where reef communities and highest marine biodiversity were concentrated, exceeded the thermal tolerance thresholds of the dominant marine invertebrate groups. Ocean warming affects marine oxygen content through a direct physical mechanism that requires no biological mediation. The solubility of oxygen in seawater decreases as temperature increases, a relationship described precisely by Henry's law and quantified across the relevant temperature range by extensive experimental data. For every degree Celsius of warming, seawater at saturation holds approximately 2% less dissolved oxygen across the warming interval reconstructed for the end Permian Ocean. Potentially 8 to 15 degrees Celsius in tropical surface waters. This solubility reduction alone would have substantially decreased the oxygen content of surface waters. The reduction occurred even before any biological or circulation-related effects are considered. In deeper waters, where oxygen is replenished only through the sinking of cold, oxygen-rich surface water from high latitudes, the consequences of warming were more severe still. The deep ocean circulation system that delivers oxygen to intermediate and abyssal depths operates through thermohaline convection, a process driven by the density contrast between cold, saline water at high latitudes and warmer, fresher water at lower latitudes. Cold surface water at the poles sinks, carrying dissolved oxygen into the deep ocean, where it spreads laterally along density surface toward lower latitudes and gradually rises back toward the surface through slow upwelling. This circulation system, operating on timescales of centuries to millennia, maintains the oxygen content of the deep ocean at levels sufficient to support benthic biological communities. When polar regions warm, reducing the temperature contrast that drives sinking, the circulation slows or stagnates. As circulation weakens, the supply of oxygen to deep waters is reduced or interrupted. Geochemical evidence from end Permian marine sediments documents this process with considerable precision. Iron speciation data. The ratio of highly reactive iron to total iron in fine-grained marine sediments, a proxy for the redox state of bottom waters during deposition, shows a transition from oxic to anoxic and in some cases euxinic conditions in deep water sections across the Permian-Triassic boundary. Euxinic conditions, in which not merely oxygen but sulfate is depleted and hydrogen sulfide accumulates in the water column, represent the most extreme end of marine oxygen depletion and are directly lethal to virtually all metazoan life. Sulfur isotope data from boundary sections, combined with the iron speciation proxy, indicates that euxinic water masses expanded significantly during the extinction interval, rising from deep basin settings towards shallower depths, where the majority of marine biodiversity was concentrated. Trace element geochemistry provides additional confirmation. Molybdenum, uranium, and vanadium are enriched in sediments deposited under anoxic conditions because they are reduced from soluble to insoluble forms in the absence of oxygen and precipitate onto the seafloor. Boundary sediments from multiple Tethyan and Panthalassan sections show enrichment in these elements consistent with widespread seafloor anoxia during the extinction interval. The global extent of these enrichments, documented in sections from China, Iran, Japan, and the Arctic, implies that anoxic conditions were not confined to isolated basins, but represented a widespread feature of the end Permian across multiple geographic regions and depositional settings. The expansion of anoxic and euxinic water masses into previously oxygenated environments had direct and immediate consequences for the biological communities inhabiting those environments. Marine invertebrates, brachiopods, bryozoans, echinoderms, mollusks that had evolved under oxygenated conditions possessed no physiological mechanisms for tolerating sustained oxygen depletion. Their metabolic requirements, calibrated by evolution to the oxygen availability of a well-ventilated Paleozoic ocean, could not be met in waters where dissolved oxygen concentrations dropped below the critical thresholds, typically in the range of 1 to 2 milligrams per liter. Below these thresholds, aerobic metabolism becomes unsustainable. Death in such conditions was not immediate in all cases, but it was inevitable, and it was spatially extensive in proportion to the area of seafloor and water column affected by deoxygenation. The reef communities of the Tethyan margin, already experiencing thermal stress from rising sea surface temperatures, faced the additional pressure of changing ocean chemistry affecting calcification. The dissolution of carbon dioxide into seawater produces carbonic acid, which dissociates to release hydrogen ions, reducing ocean pH and decreasing the concentration of carbonate ions available for shell and skeleton formation. This process, ocean acidification, reduces the saturation state of seawater with respect to the calcium carbonate minerals, calcite and aragonite, from which marine invertebrates construct their hard parts. Evidence for end Permian ocean acidification is preserved in the skeletal geochemistry of marine fossils from boundary sections. Boron isotope data from carbonate fossils, a proxy for seawater pH at the time of shell formation, shows a negative excursion across the Permian-Triassic boundary. The excursion is consistent with a pH reduction of approximately 0.1 to 0.7 units, depending on the section and the calibration applied. This range of acidification would have been sufficient to significantly impair calcification in organisms near the lower limits of their carbonate saturation tolerance. These organisms were already living in waters warmed above their optimal thermal range. The simultaneous application of thermal stress, oxygen depletion, and acidification to marine communities that had no evolutionary preparation for any of these conditions in combination produced a mortality pattern consistent with the fossil record. The most severe losses occurred among sessile, reef-associated filter feeders, brachiopods, bryozoans, crinoids, and sponge reef framework builders that were unable to relocate in response to deteriorating local conditions. Their physiological tolerance ranges were among the narrowest. Mobile organisms with broader physiological flexibility, certain fish lineages, some mollusks, and various microbial communities showed higher survival rates. This pattern is consistent with the selective pressure of environmental stressors that rewarded physiological generalism over ecological specialization. The deep ocean, deprived of oxygen and increasingly dominated by anaerobic microbial communities, metabolizing sulfate and producing hydrogen sulfide as a byproduct, underwent a fundamental ecological transformation. The benthic communities that had inhabited deep and intermediate water depths across the Paleozoic, diverse assemblages of deposit feeders, filter feeders, and their predators, were replaced by microbial mats and chemosynthetic communities tolerant of sulfidic conditions. Biotubation was replaced by finely laminated sediment characteristic of anoxic bottom waters where no burrowing metazoans survived. The oceans that had taken the entirety of the Paleozoic to populate with biological complexity were being emptied, not instantaneously and not uniformly across all regions simultaneously, but systematically. Depth by depth, habitat by habitat, the chemical conditions sustaining life contracted toward a narrower and narrower range. Concentrated in the shallowest, best ventilated surface waters, where the last refugia of marine diversity would persist through the worst of what was coming. The end Permian extinction was not instantaneous. The geological and biological evidence preserved across Permian-Triassic boundary sections on multiple continents and in multiple ocean basins describes a process that, while catastrophically rapid by geological standards, unfolded across a time frame measured in thousands to tens of thousands of years. The end Permian extinction was not instantaneous. The geological and biological evidence preserved across Permian-Triassic boundary sections on multiple continents and in multiple ocean basins describes a process that, while catastrophically rapid by geological standards, unfolded across a time frame measured in thousands to tens of thousands of years. The process lasted long enough for the progressive dismantling of ecological structure to proceed through identifiable stages, each compounding the vulnerability of what remained. The distinction between a sudden extinction and a prolonged one is not merely semantic. It determines the mechanisms through which biological communities collapse, the sequence in which different ecological components fail, and the degree to which survival becomes possible for any lineage at all. The earliest phase of biological deterioration on land is recorded in the vegetation record. Palynological sequences, the stratigraphic record of pollen and spores preserved in fine intercepts from Permian-Triassic boundary sections in China, Greenland, Russia, and Australia, document a collapse in plant diversity. The collapse preceded or coincided with the first major losses in vertebrate fauna. The transition is characterized by a sharp reduction in gymnosperm pollen, representing the dominant forest trees of the late Permian, and a corresponding increase in spores of lycopsids, ferns, and other opportunistic, stress-tolerant plant groups. In some boundary sections, this interval is marked by what palynologists term the coal gap, a cessation of peat and coal accumulation that persists across the boundary interval and reflects the disappearance of the productive, continuously vegetated wetland ecosystems that had been generating organic accumulations throughout the Permian. The loss of forest cover initiated a cascade of consequences, operating through multiple interconnected pathways simultaneously. Vegetation stabilizes soils through root binding and the maintenance of organic matter content that preserves soil structure against erosional forces. When plant cover is removed or severely reduced, soils become vulnerable to erosion by rainfall and wind at rates orders of magnitude higher than those operating under vegetated conditions. Sedimentological evidence from Permian transboundary sandkeys sequences in multiple continental basins documents a transition to high-energy, coarse-grained fluvial deposits at the boundary interval. This represents a shift from the fine-grained, organic-rich sediments characteristic of stable, vegetated landscapes to the poorly sorted, erosional deposits characteristic of landscapes stripped of stabilizing vegetation cover. In some sections, this transition occurs within a stratigraphic interval representing as little as a few thousand years. The erosion of soils released into river systems and ultimately into the ocean, the nutrient and mineral content accumulated across millions of years of biological activity. This nutrient pulse, phosphorus, nitrogen compounds, and dissolved minerals stimulated primary productivity in coastal marine environments through a process analogous to modern eutrophication events. The blooms of algae and cyanobacteria produced by this nutrient influx consume dissolved oxygen as they decomposed, further expanding the oxygen-depleted zones already developing through warming and circulation slowdown. The terrestrial and marine systems, already stressed by independent forcing from volcanic emissions, were connected through this nutrient pathway in a feedback that amplified deteriorations in both simultaneously. For terrestrial animal communities, the collapse of vegetation represented the removal of the foundational resource upon which all higher trophic levels depended. The herbivore populations of the Permian, dicynodonts, pariasaurs, and the diverse array of smaller plant-consuming therapsids were dependent on the continuous productivity of Permian plant communities for the caloric input required to sustain their population densities. As plant communities simplified, contracted, and in some regions disappeared entirely, the carrying capacity of terrestrial landscapes for herbivorous animals declined in proportion. Population sizes fell, geographic ranges contracted toward refugia where plant productivity persisted. Reproductive success is decreased as nutritional stress reduced the physiological condition of breeding adults and increased juvenile mortality. The predator communities dependent on herbivore populations experienced these effects at one remove. The effects were delayed, but equally severe in terms of long-term population viability. Gorgonopsian predators, whose population densities were already constrained by the energetic inefficiency of carnivory relative to herbivory, required large and stable prey populations to sustain viable breeding groups. As dicynodont and other herbivore populations declined, the prey base supporting apex predators contracted below the threshold required to maintain predator populations at reproductive viability. The fossil record of Gorgonopsians does not extend past the Permian-Triassic boundary in any section yet analyzed. They disappear entirely, representing one of the most ecologically significant losses of the extinction event. Their extinction eliminated an entire functional predator guild from the terrestrial ecosystem. The temporal structure of extinction across the boundary interval is recorded with particular resolution in the Karoo Basin of South Africa. Continuous sedimentation across the boundary and high fossil density allow the disappearance of individual taxa to be tracked stratigraphically. Analysis of multiple Karoo boundary sections indicates that vertebrate species loss was not uniformly distributed across the boundary interval. Instead, the fossil record shows a pattern of accelerating loss, initial reductions in diversity, giving way to more rapid disappearance rates as the boundary horizon is approached. This acceleration is consistent with a system in which early losses destabilize ecological structure, increasing the vulnerability of surviving species to subsequent stressors through the removal of ecological partners, prey, or habitat engineers upon which their survival indirectly depended. The concept of ecological cascade, in which the loss of one species or functional group increases the extinction probability of others connected to it through food web relationships or ecosystem engineering, is well documented in modern conservation biology. It provides a framework for interpreting the progressive character of the end Permian terrestrial extinction. The disappearance of large herbivores reduces vegetation control, potentially altering plant community composition in ways that disadvantaged specialists adapted to particular plant types. The loss of burrowing animals reduces soil aeration and mixing, affecting plant root environments and soil invertebrate communities. The elimination of apex predators releases mesopredator populations from top-down control. This release can drive prey species to local extinction through unsustainable predation pressure, before the mesopredators themselves collapse for lack of resources. Each of these mechanisms operates through ecological relationships built across millions of years. Each mechanism amplifies the mortality signal initiated by the primary environmental forcing. In the marine realm, the prolonged character of the extinction is recorded in the stratigraphic distribution of last occurrences of marine taxa across boundary sections of varying sedimentation rate. High-resolution sections from South China, where carbonate accumulation rates during the boundary interval are sufficient to resolve changes at centennial to millennial timescales, show that marine invertebrate diversity declined through a series of steps rather than a single instantaneous collapse. Individual genera disappear at different stratigraphic levels within the boundary interval, with some taxa persisting through the initial phases of environmental deterioration before disappearing at later horizons. These later extinctions occurred when conditions worsened further, or when the prey and habitat resources they depended upon were finally eliminated. The selectivity of survival during this prolonged dying phase provides information about which biological characteristics conferred resilience under end Permian conditions. Small body size, reducing absolute metabolic requirements and allowing population maintenance on reduced resource availability, appears to correlate with higher survival probability across multiple taxonomic groups. Physiological generalism, tolerance of a wider range of temperatures, oxygen levels, and food sources similarly correlates with persistence through the boundary interval relative to specialists with narrow tolerance ranges. Mobility, the capacity to relocate in response to locally deteriorating conditions, provided an advantage over sessile organisms unable to track shifting environmental gradients. These selective filters, operating across thousands of years of progressive deterioration, shaped the composition of the post-extinction fauna in ways that would define the trajectory of life's recovery across the subsequent millions of years. The survivors were not the largest, the most ecologically dominant, or the most anatomically sophisticated organisms of the late Permian world. They were those whose biological characteristics happened to align with the specific demands of surviving a prolonged, multi-stressor environmental collapse. A selection pressure unlike any that had operated across the preceding Paleozoic. That pressure would leave a permanent imprint on the evolutionary lineages that emerged from it. The dying was long, and for the ecosystems of the Permian world, that length was not a mercy. It was a mechanism, one that dismantled biological complexity layer by layer, removing each structural component in a sequence determined not by evolutionary fitness in any stable sense, but by the order in which the environmental conditions sustaining each component failed. The progressive deterioration of atmospheric chemistry, ocean circulation, and terrestrial vegetation documented across the Permian-Triassic boundary interval produced, at its culmination, a set of surface conditions that represented a fundamental departure from anything the living world had experienced within the Paleozoic. The planet had not simply become less hospitable. It had, across a geologically brief interval, been transformed into an environment actively hostile to the biological systems that had developed within it. One in which the strict physical and chemical parameters sustaining complex life were simultaneously and persistently outside the ranges those systems required to function. Temperature is the most directly measurable of these parameters, and the thermal record of the end Permian interval is among the most thoroughly documented aspects of the extinction event. Oxygen isotope data from conodont apatite, the mineralized tooth elements of small eel-like marine animals that are among the most reliable records of seawater temperature in the Permian-Triassic boundary interval, from sections in South China and the Tethyan region, show a temperature excursion of extraordinary magnitude centered on the boundary horizon. Published analyses of these records indicate tropical sea surface temperatures reaching 35 to 40 degrees Celsius during the peak warming interval, values that approach or exceed the upper thermal tolerance limits of virtually all complex marine organisms under sustained exposure. The significance of these temperatures requires context. Modern tropical marine ecosystems, which represent the most thermally stressed of current ocean environments, operate at sea surface temperatures typically between 28 and 30 degrees Celsius. The physiological processes of tropical marine invertebrates, protein synthesis, enzyme function, reproductive biology, and cellular membrane integrity, are calibrated to this range through evolutionary adaptations spanning tens of millions of years. Above approximately 32 to 34 degrees Celsius, sustained for periods of weeks to months, most tropical marine invertebrates experience protein denaturation, metabolic failure, and reproductive collapse, a condition observable today in coral bleaching events driven by anomalous warming of 1 to 2 degrees above seasonal maxima. The temperatures reconstructed for the end Permian Ocean exceeded these modern stress thresholds by margins of 5 to 10 degrees Celsius, sustained not for weeks, but across intervals reconstructed at thousands to tens of thousands of years. Terrestrial temperatures during the peak end Permian warming interval are less precisely constrained than marine values, owing to the greater difficulty of extracting quantitative temperature signals from continental depositional environments. However, paleosol carbonate isotope data, plant fossil assemblage analysis, and climate model outputs forced with end Permian boundary conditions consistently indicate continental interior temperatures substantially elevated above late Permian baselines. In low-latitude continental interiors, regions already subject to elevated temperatures under the geography of Pangaea, with its vast landmass far from oceanic thermal buffering, peak warming may have pushed mean annual temperatures above 50 degrees Celsius in some regions, according to climate model simulations constrained by available proxy data. Temperatures of this magnitude are physiologically lethal to most terrestrial vertebrates under sustained exposure, and would have rendered large portions of equatorial Pangaea biologically uninhabitable regardless of food or water availability. The thermal gradient between equatorial and polar regions, the primary driver of atmospheric circulation, and the wind systems that distribute moisture across continental surfaces, was reduced by end Permian warming as high-latitude regions warmed proportionally more than low-latitude ones. This reduction in the equator to pole temperature gradient weakened the Hadley circulation and the jet stream systems that drive precipitation patterns across continental interiors. Climate model simulations of end Permian conditions consistently produce a collapse of the monsoon systems that had delivered seasonal rainfall to large portions of Pangaea during the late Permian, replacing them with persistent aridity across continental regions that had previously received sufficient precipitation to support diverse biological communities. The hydrological consequences of this circulation change compounded the direct thermal stress. river systems dependent on seasonal precipitation, the primary water source for terrestrial ecosystems far from coastlines, experienced reduced and increasingly unreliable discharge. Paleosoil sequences from Permian-Triassic boundary sections in the Karoo Basin and in continental deposits from China and Russia document a transition to increasingly arid soil-forming conditions across the boundary interval, with calcrete and evaporite mineral formation indicating moisture deficits inconsistent with the maintenance of productive vegetation or the survival of large-bodied vertebrates dependent on permanent water sources. Some river systems, based on sedimentological evidence of their depositional behavior, appear to have shifted from perennial to ephemeral flow, carrying water only during infrequent precipitation events and remaining dry for extended intervals. A transition that would have severed the connectivity between aquatic refugia, upon which both aquatic and terrestrial communities depended. The interactions between elevated temperature and reduced moisture availability produced a fire regime on the late Permian landscape for which the Permian world had no precedent. Charcoal, fossil charred plant material, termed fusane in the geological literature, is preserved in sedimentary sequences from multiple continents across the Permian-Triassic boundary interval, indicating widespread burning of terrestrial vegetation. While charcoal is present in pre-extinction Permian sequences as well, reflecting normal background fire occurrence in an oxygen-rich atmosphere, its abundance and geographic distribution increases significantly in boundary and early Triassic sequences, consistent with more frequent and extensive fire activity under the combined conditions of elevated temperature, increased aridity, and accumulation of dead plant material from dying forests. Fire, operating as a direct mortality agent and as a mechanism for accelerating soil loss and carbon release, added a further destabilizing pressure to terrestrial ecosystems already compromised by thermal and moisture stress. The freshwater systems of the Permian world, rivers, lakes, and wetlands that had sustained diverse communities of fish, amphibians, and invertebrates across the Paleozoic, were among the most severely affected terrestrial environments during the peak extinction interval. Freshwater systems are thermally sensitive environments, and the warming documented across the boundary interval would have driven water temperatures in shallow lakes and slow-moving river systems toward values lethal to the cold water and temperate fish lineages that had dominated Permian freshwater communities. Simultaneously, increased weathering of exposed soils, accelerated by acid precipitation and the loss of vegetation, delivered elevated concentrations of dissolved metals and altered nutrient loads into freshwater systems, further degrading water chemistry. The amphibian communities dependent on fresh water for reproduction faced the compounded stress of extremes. Altered water chemistry and the progressive disappearance of the invertebrate prey communities upon which larval and adult amphibians fed. Oxygen availability in the terrestrial atmosphere during the end Permian interval has been modeled using carbon and sulfur cycle reconstructions constrained by isotopic data as declining from the elevated late Permian values near or above 30% by volume toward values closer to modern concentrations of approximately 21% or potentially lower during the peak boundary interval. This decline, driven by the collapse of terrestrial plant productivity and the reduction in organic carbon burial that had maintained elevated Permian oxygen levels, would have reduced the physiological capacity of large-bodied terrestrial animals dependent on high oxygen availability for sustained activity. For the giant insects whose body size was sustained by the oxygen-rich Permian atmosphere, the decline toward lower oxygen concentrations represented an additional physiological stress, compounding the thermal and nutritional pressures already reducing their populations. The geological record of the end Permian boundary interval describes a planet that had, through the cumulative interaction of volcanic forcing, atmospheric chemistry change, ocean deoxygenation, terrestrial vegetation collapse, and thermal extremes, arrived at a state in which the basic physical requirements for complex life, moderate temperatures, available water, oxygenated atmosphere and ocean, functional food webs were simultaneously compromised across the majority of the planet's surface. This was not a single catastrophe superimposed on a functioning world. It was the end state of a progressive destabilization in which each component failure increased the severity of the others through interconnected feedback pathways, producing a compound environmental crisis with no precedent in the biological experience of any organism alive at the time. The world that had sustained the complexity of the Permian, its forests, its reefs, its diverse predator guilds and intricate marine communities, had not simply changed. It had, across a geological interval of extraordinary brevity, become something that most of the life it had spent 300 million years producing could no longer survive within. What followed was not a transition, it was a reckoning. The term applied in the scientific literature to the end Permian extinction event, the Great Dying, is one of the few instances in geological nomenclature where the descriptive language approaches the actual scale of what it names. The extinction that peaked at the Permian-Triassic boundary, approximately 252 million years ago, represents the largest recorded loss of biological diversity in the history of complex animal life on Earth. Its magnitude, measured across multiple independent proxies and confirmed through decades of stratigraphic, paleontological, and geochemical analysis across dozens of boundary sections on multiple continents, places it in a category without parallel in the Phanerozoic record, a biological catastrophe so comprehensive that its effects reorganized the trajectory of evolution on this planet for the subsequent 50 million years. The quantification of extinction magnitude at the Permian-Triassic boundary has been approached through multiple methodological frameworks, each with its own sources of uncertainty and its own strengths. The most commonly cited figure, that approximately 90 to 96% of all marine species and approximately 70% of terrestrial vertebrate species were lost, derives from compilations of fossil occurrence data across boundary sections combined with statistical methods designed to correct for the uneven preservation and sampling of the fossil record. These figures have been refined repeatedly as new boundary sections have been analyzed and as statistical methods have improved. But the broad magnitude of the estimate has remained consistent across several decades of research and across the independent analyses of multiple research groups using different datasets and methodologies. The marine extinction record is the more precisely documented of the two, owing to the greater density and continuity of marine fossil sequences across the boundary interval relative to continental deposits. Within the marine realm, the losses were not distributed uniformly across taxonomic groups or ecological categories. Sessile, reef-associated filter feeders, the brachiopods, bryozoans, crinoids, and calcareous sponges that had dominated shallow marine communities throughout the Paleozoic, suffered the most severe losses, with some groups losing in excess of 95% of their species diversity across the boundary interval. The rugus and tabulate corals, the dominant reef framework builders of the Paleozoic, did not survive the extinction at all, disappeared entirely from the fossil record at the boundary horizon and leaving the reef-building ecological role vacant for approximately 10 years into the Triassic, before scleractinian corals, representing an entirely new evolutionary lineage, began to fill it. Trilobites, which had persisted through multiple previous extinction events across more than 270 million years of evolutionary history, did not survive the end Permian event. Their last occurrences in the fossil record are documented in latest Permian sediments. No trilobite fossils have been recovered from any Triassic age deposit worldwide, despite extensive sampling of early Triassic marine sequences. Their disappearance represents the termination of one of the longest-running and most morphologically diverse animal lineages in the Paleozoic fossil record, a group that had occupied marine environments continuously since the Cambrian. surviving the late Ordovician, late Devonian, and end Carboniferous extinction pulses, only to be eliminated entirely by conditions at the Permian-Triassic boundary. Foraminifera, the single-celled organisms whose shells provide some of the most detailed geochemical records of boundary conditions, experience species-level losses approaching 90% across the boundary interval in the most thoroughly sampled Tethyan sections. The fusolinid foraminifera, a group characterized by multi-chambered shells that had diversified enormously during the Carboniferous and Permian, and whose remains constitute a significant fraction of many Permian limestone sequences, disappeared entirely at the boundary, their extinction eliminating a major component of marine carbonate production that had been operating continuously for more than 80 million years. Ammonoid cephalopods, which had recovered from a near-extinction event earlier in the Permian and diversified considerably in the period's final stages, were reduced to a single or very small number of surviving lineages at the boundary, from which all Triassic and later Mesozoic ammonoid diversity would subsequently radiate. The bottleneck imposed by the end Permian extinction on this group was severe enough that early Triassic ammonoid assemblages show dramatically reduced morphological disparity relative to their late Permian predecessors, reflecting the limited genetic diversity available to the founding survivor populations. Their subsequent diversification across the Triassic, eventually producing the extraordinary diverse Mesozoic ammonoid fauna, demonstrates the evolutionary potential that even severely bottlenecked survivor lineages retain, given sufficient time and ecological opportunity. The terrestrial vertebrate record at the boundary presents a pattern of loss consistent with, though less precisely quantified than, the marine record. The therapsid fauna that had dominated Permian land ecosystem's Gorgonopsians, most dicynodont lineages, the majority of therocephalian and parareptilian diversity, disappears from the fossil record at or before the boundary horizon in the most thoroughly sampled continental sections. The Karoo Basin assemblage zones, which provide the most continuous record of late Permian terrestrial vertebrates available, document a transition from diverse multi-taxon communities in the latest Permian to dramatically impoverished early Triassic assemblages, dominated by a small number of surviving dicynodont and cynodont lineages. The ecological complexity of the late Permian terrestrial fauna, its multiple predator guilds, its diverse herbivore assemblages, its array of ecological specialists adapted to particular habitat types, was reduced across the boundary interval to a simplified assemblage of generalist survivors occupying a landscape stripped of the ecological structure that had previously supported greater diversity. The selectivity of the end Permian extinction, while less pronounced than in some other extinction events, is nonetheless discernible in the fossil record. Ecological generalists, organisms capable of tolerating a wider range of environmental conditions and exploiting a broader range of food sources, show higher survival rates across multiple taxonomic groups than specialists. Small body size correlates with higher survival probability in terrestrial vertebrates, consistent with the lower absolute resource requirements of smaller animals in a world of reduced productivity. In the marine realm, organisms with planktonic larval stages capable of dispersing across ocean bed to locate surviving harpy habitat show higher survival rates than purely benthic organisms with limited dispersal capacity. These selective patterns are consistent with the nature of the end Permian stressors. Thermal extremes, resource depletion, and habitat destruction, and with the physiological and ecological characteristics that would have conferred resilience under those specific conditions. The geographic pattern of extinction intensity across the boundary is not uniform, and its variation provides additional information about the mechanisms driving mortality. Tropical marine environments, already operating near the upper thermal limits of their constituent organisms, show higher extinction intensities than higher latitude sections in most compilations of boundary fossil data consistent with the lethal ocean temperature reconstructions from low-latitude sections. Deep-water marine environments, subjected to the most severe deoxygenation, show near-complete faunal replacement across the boundary. High-latitude terrestrial environments, potentially buffered from the most extreme thermal excursions by their distance from the equator, may have served as refugia for some lineages that disappeared from lower latitude sections earlier in the boundary interval. The duration of peak extinction, the interval during which the highest rates of species disappearance are concentrated, has been estimated from high-resolution boundary sections in South China at approximately 60,000 years or less, based on uranium lead dating of volcanic ash beds bracketing the main extinction horizon. This figure, while representing a geological instant, encompasses a time frame during which thousands of generations of most animal species would have lived and died. Sufficient for population-level responses, including range contraction, behavioral modification, and limited phenotypic plasticity, but insufficient for the evolutionary generation of new adaptive solutions to novel environmental conditions. What the combined fossil, geochemical, and geochronological record of the Permian-Triassic boundary describes is not simply the death of species, it is the collapse of ecological architecture. The simultaneous elimination of the functional roles, trophic relationships, and environmental engineering capacities that complex biological communities require to sustain themselves. Individual species can be replaced through evolution across millions of years. The integrated ecological structure that took the entirety of the Paleozoic to construct, its food webs, its biogeochemical cycles, its mutually sustaining relationships between organisms and environments, cannot be reconstructed quickly. Its loss constituted a biological impoverishment of the planet that would persist measurably for millions of years into the Triassic. The Great Dying was not the end of life on Earth, but it came closer to that outcome than any other event in the history of complex animals. And the world it left behind was, in almost every measurable biological sense, a different planet from the one that had existed before it began. The Permian-Triassic boundary represents, in the fossil record, one of the sharpest discontinuities in the history of complex life. Below the boundary horizon, in the latest Permian sediments, the diverse and ecologically structured communities of the Paleozoic world are documented in their full complexity. Above it, in the earliest Triassic, the fossil record presents a world of dramatically reduced biological diversity, simpler in ecological structure, lower in species richness, and dominated by a small number of lineages whose survival through the boundary interval was determined by a combination of physiological characteristics, geographic circumstance, and the specific nature of the stressors that eliminated the majority of their contemporaries. Understanding which organisms survived, why, requires examining both the biological characteristics of the survivors themselves and the conditions under which survival was possible. The most fundamental characteristic shared across surviving lineages in both marine and terrestrial environments is physiological breadth, the capacity to tolerate a wider range of environmental conditions than the specialized, ecologically refined organisms that dominated late Permian communities. This characteristic is not a measure of biological sophistication. In many cases, the survivors of the end Permian extinction were less anatomically complex, less ecologically specialized, and less physiologically refined than the organisms they outlasted. Their survival reflected not competitive superiority in stable conditions, but resilience under conditions of extreme and sustained environmental instability, a fundamentally different selective environment from that which had shaped the Permian world across the preceding millions of years. In the marine realm, the organisms that persisted through the boundary interval and into the earliest Triassic represent a taxonomically restricted and ecologically simplified subset of late Permian marine diversity. Certain bivalve mollusks, including members of the genus Claria, whose thin, flat shells and tolerance of low oxygen conditions appear in early Triassic sediments across the Tethyan region in large numbers immediately above the extinction horizon, survived and expanded rapidly into the ecological space vacated by the collapse of Paleozoic marine communities. Their abundance in earliest Triassic sections is in some ways misleading. It reflects not biological recovery in any meaningful ecological sense, but the opportunistic proliferation of stress-tolerant generalists into a world from which their competitors and predators had been largely removed. Claria assemblages in early Triassic sections often show low species diversity and high numerical dominance, the biological signature of a disturbed system colonized by opportunists rather than a recovering one rebuilding ecological complexity. Gastropod mollusks similarly showed higher survival rates than many other marine invertebrate groups, consistent with their broader physiological tolerance ranges and their capacity for both benthic and semi-pelagic lifestyles that allowed exploitation of the limited habitable zones remaining in a deoxygenated ocean. Some fish lineages persisted through the boundary interval, their higher mobility and physiological flexibility relative to sessile invertebrates, allowing them to track the contracted zones of oxygenated, thermally tolerable water that persisted in higher latitude or deeper mixed layer-layer ocean environments. Early actinopterygian fish, the ray-finned lineage that would eventually diversify into the dominant fish group of all subsequent geological periods, are represented in earliest Triassic marine deposits, their survival initiating a diversification trajectory that would, across the subsequent 50 million years, produce the ecological dominance they maintain in modern oceans. Microbial communities, whose metabolic versatility and tolerance extreme conditions far exceeds that of any multicellular organism, not only survived the end Permian extinction, but expanded dramatically in its immediate aftermath. Microbialites, carbonate structures produced by the metabolic activity of microbial mats, predominantly cyanobacteria, appear in earliest Triassic shallow marine sections from multiple regions, including the Tethyan margin and western North America, in a distribution and abundance unprecedented in the preceding late Permian record. Their proliferation represents the filling of shallow marine substrate by microbial communities in the absence of the grazing, burrowing, and competitive pressure normally exerted by metazone organisms. The temporary dominance of microbialites, earliest Triassic marine environments, is one of the most visually distinctive features of post-extinction ocean communities and has been interpreted as evidence for the near-complete collapse of the metazone ecological framework that had suppressed microbial mat expansion throughout the Paleozoic. On land, the terrestrial vertebrate survivors of the end Permian extinction represent a similarly restricted and ecologically simplified assemblage relative to the late Permian fauna they replaced. Among therapsids, the lineage that showed the highest survival rate through the boundary interval was the Dicynodonts, specifically a subset of Dicynodont diversity, represented by generalist, medium-bodied forms capable of exploiting a range of plant material and tolerating the degraded vegetation communities of the earliest Triassic. Lystrosaurus, a Dicynodont genus whose remains are among the most abundant of any terrestrial vertebrate in earliest Triassic continental deposits worldwide, exemplifies the survivor profile. It was not the largest, most ecologically dominant, or most anatomically specialized of late Permian Dicynodonts. It was, based on the evidence of its skeletal anatomy and its geographic distribution across multiple paleolatitudes Permian, a physiological generalist, capable of tolerating a range of temperatures, exploiting low-quality food sources, and surviving in degraded environments where more specialized contemporaries could not persist. The dominance of Lystrosaurus in earliest Triassic continental deposits is quantitatively remarkable. In some Karoo Basin early Triassic sections, Lystrosaurus constitutes more than 90% of all vertebrate fossil specimens recovered. a proportion of numerical dominance that has no equivalent in any normal ecologically diverse vertebrate fossil assemblage, and that reflects the near-total elimination of competing lineages rather than any exceptional biological prowess of the animal itself. Its geographic distribution in earliest Triassic deposits spans what are now Antarctica, South Africa, India, China, and Russia. Essentially the full extent of the surviving Pangaean landmass for which early Triassic continental deposits have been sampled, indicating that its survival was not geographically restricted to a single refugium, but extended across a broad range of continental environments that had been simplified to a degree that this generalist could exploit. Cynodont therapsids, though less numerically dominant than Lystrosaurus in earliest Triassic assemblages, represent a survivor lineage of disproportionate long-term evolutionary significance. The cynodonts that crossed the Permian-Triassic boundary were small, most genera estimated at body masses below 1 kilogram. Insectivorous or omnivorous animals whose low absolute resource requirements allowed population maintenance in environments of severely reduced productivity. Their skeletal anatomy shows progressive development toward the mammalian body plan, including evidence for a more fully upright posture, more complex dentition, allowing efficient food processing, and skull anatomy consistent with higher metabolic rates than earlier therapsid lineages. These characteristics, which had evolved under the competitive pressures of the late Permian ecosystem, proved to be precisely the suite of adaptations required for survival in the resource-depleted environments of the earliest Triassic. The geographic distribution of survival across the boundary interval was not random. Evidence from multiple boundary sections suggests that higher latitude environments, removed from the most extreme thermal conditions of the equatorial regions, served as refugia in which some lineages persisted through the peak extinction interval, before subsequently expanding into lower latitude environments as conditions gradually moderated. High-latitude marine section shows somewhat lower extinction intensities than tropical sections in some analyses, consistent with a thermal refugium effect. Continental high-latitude deposits from what is now Antarctica preserve early Triassic vertebrate faunas that, while impoverished relative to the late Permian, retain somewhat higher diversity than some low-latitude sections, further supporting that geographic thermal gradient influence survival probability at the population level. What the survivor record ultimately describes is not a random sample of late Permian biodiversity preserved by chance. It is a filtered subset, selected by the specific character of the Permian stressors for physiological breadth, small body size, low resource requirements, and geographic position in less severely affected regions. The biological world that would emerge from this filter, built from the genetic and ecological raw material of these survivors, would be fundamentally shaped by the characteristics of the lineages that crossed the boundary, and by the absence of the lineages that did not. The Paleozoic was over. What came next would have to be built from what remained. The earliest Triassic period, spanning approximately the first 5 million years following the Permian-Triassic boundary, represents one of the most biologically impoverished intervals in the history of complex animal life. The geological and paleontological record of this interval describes a planet that had not simply lost species, but had lost the ecological architecture within which species exist. The trophic relationships, habitat structures, biogeochemical cycles, and mutually sustaining interactions between organisms and environments that constitute a functioning biosphere. What remained was not a simplified version of the Permian world. It was something categorically different. A planetary surface in which the biological systems that had regulated chemistry, stabilized landscapes, and sustained productivity across the Paleozoic were either absent or operating at a fraction of their former capacity. The duration of this biological impoverishment, the interval during which recovery was minimal, and ecosystems remained in a simplified, disturbed state, has been estimated from the fossil record and from geochemical proxies at between 4 and 9 million years, with significant regional variation in both the severity of initial impoverishment and the timing of measurable recovery. This figure is itself a measure of the depth of the extinction's impact. Previous extinction events in the Paleozoic record, including the late Ordovician and late Devonian extinctions, both of which produced significant biodiversity loss, were followed by measurable biological recovery within 1 to 3 million years. The extended duration of post-Permian biological impoverishment reflects the exceptional severity of the end Permian event and the degree to which it disrupted not just species diversity, but the ecological and geochemical processes upon which recovery depends. The marine fossil record of the earliest Triassic is characterized by what paleontologists term the survival interval, a period in which diversity remained severely depressed and community composition was dominated by a small number of opportunistic, stress-tolerant taxa. The abundant chloria, bivalve assemblages of the earliest Triassic, present in sections across the Tethyan region, and in deposits from what is now British Columbia, Nevada, and the Arctic, represent this survival fauna in its most numerically visible expression. These assemblages are ecologically simple, dominated by a single genus or a small number of related forms, lacking the multi-trophic structure of pre-extinction marine communities, and showing little of the morphological and ecological diversity that characterizes recovered marine ecosystems in later Triassic sections. The carbon isotope record of earliest Triassic marine carbonates shows persistent instability throughout the survival interval. Repeated negative excursions indicate continuing disruption of the global carbon cycle for millions of years following the main extinction horizon. These excursions, documented in sections from China, Italy, and the Arctic, reflect ongoing perturbation of the biological and geochemical processes that regulate the carbon cycle under normal conditions. Primary productivity in the surface ocean, the photosynthetic fixation of carbon dioxide by phytoplankton and algae that forms the base of marine food webs, remained suppressed throughout the early Triassic survival interval, as indicated by low organic carbon burial rates and isotopic signatures inconsistent with high biological productivity. The ocean's capacity to sustain complex food webs was constrained at its foundation, limiting the rate at which higher trophic levels could recover, even as environmental conditions gradually improved. Reef ecosystems, among the most structurally complex and biologically diverse of all marine habitats, and among the most severely affected by the end Permian extinction, show one of the longest recovery delays of any marine ecological system in the Triassic record. The disappearance of rugose and tabulate corals at the Permian boundary left the reef-building ecological role vacant for an extended interval. Earliest Triassic shallow marine carbonate sections that would, in a normal recovery scenario, be expected to show the beginnings of reef framework construction instead, document microbialite-dominated substrates, thin carbonate crusts produced by abiotic precipitation, and sparse early Metazone colonization without the structural complexity of true reef frameworks. Quantitative analyses of reef occurrence in Triassic marine sections indicate that ecologically significant Metazone reef structures do not reappear in the fossil record until approximately 8 to 10 million years after the extinction horizon, one of the longest reef gaps in the entire Phanerozoic record. The terrestrial record of the earliest Triassic presents a landscape of comparable biological simplicity. Palynological sequences from early Triassic continental deposits across multiple continents document a prolonged interval of low plant diversity, dominated by fern spores and lycopsid spores characteristic of opportunistic, early colonizing plant communities rather than the diverse gymnosperm-dominated forests of the Permian. This interval, sometimes termed the fern spike, by analogy with similar post-disturbance plant community signatures documented in the geological record of other major events, reflects the colonization of denuded, erosion-prone landscapes by fast-growing, stress-tolerant plant taxa in the absence of the slower-growing but structurally dominant trees that had characterized Permian forests. The fern-dominated communities of the earliest Triassic provided ground cover and limited erosion control, but they lacked the biomass, structural complexity, and soil-building capacity of the forests they replaced. Soil development in the earliest Triassic reflects the impoverished state of terrestrial vegetation cover. Paleosol sequences from early Triassic continental deposits show thin, weakly developed soil profiles, lacking the deep organic horizons characteristic of mature, vegetated landscapes. Calcrete formation, indicating moisture deficits and limited biological soil processing, is widespread in early Triassic paleosols from multiple continents, consistent with the persistence of arid or semi-arid conditions across large areas of continental Pangaea for millions of years following the boundary. The soil ecosystems that had taken millions of years of biological activity to construct during the Permian. Their fungal networks, invertebrate communities, and organic matter stores required the restoration of surface vegetation before they could begin to rebuild, creating a recovery dependency that slowed terrestrial ecosystem reconstruction across multiple organizational levels simultaneously. The vertebrate fauna of the earliest Triassic continental environments presents a picture of extreme ecological simplification relative to the late Permian. Lystrosaurus dominated assemblages, in which a single dicynodont genus accounts for the overwhelming majority of vertebrate fossil material, persist through the earliest Triassic in the Karoo-Beocene and in correlative deposits from other continents. The ecological structure of these assemblages, lacking apex predators, dominated by a single large-bodied herbivore, with cynodont insectivores occupying the only other consistently documented vertebrate niche, represents a reduction in food web complexity to its minimum functional configuration. The multi-trophic terrestrial ecosystems of the Permian, with their apex Gorgonopsian predators, diverse herbivore guilds, and array of ecological specialists, had been compressed into a two-level system in which most of the intermediate ecological structure was absent. Freshwater environments of the earliest Triassic show patterns of biological recovery somewhat faster than marine and terrestrial systems in some regions, a pattern that has been attributed to the relative buffering of freshwater habitats from the most severe marine stresses, particularly ocean acidification and widespread deoxygenation. Temnospondyl amphibians, whose remains are documented in early Triassic freshwater deposits from multiple continents, represent one of the more ecologically intact surviving lineages, retaining their functional role as aquatic predators in river and lake systems, even as the surrounding terrestrial and marine ecosystems remained severely impoverished. Their persistence in freshwater environments may reflect the relative stability of those habitats as refugia during the peak extinction interval, and their early Triassic distributions suggest that freshwater systems served as partial reservoirs of biological diversity, from which some recovery of adjacent terrestrial communities could proceed. The silence of the earliest Triassic world was not the silence of absence alone. It was the silence of disrupted process of biogeochemical cycles operating without the biological machinery that had regulated them, of landscapes eroding without the vegetation that had stabilized them, of ocean chemistry fluctuating without the carbonate-producing and organic carbon-burying communities that had buffered it. The planet was not dead, but the systems that had made it biologically complex, that had made it, in the fullest sense, alive, were reduced to their most basic functional components, operating at the margins of viability across a surface that bore little resemblance to the world that had existed only millions of years before. Recovery, when it came, would come slowly, built upward from these remnants, one ecological relationship at a time, across millions of years of gradual biological reconstruction. The empty world of the earliest Triassic was not a permanent condition, but its emptiness was real and its duration was long enough to leave a permanent imprint on the evolutionary trajectory of every lineage that lived through it. The biological recovery from the end Permian extinction was not a restoration. The ecosystems that assembled across the Triassic period were not reconstructions of the Paleozoic world from which they emerged. They were new configurations, built from a filtered set of surviving lineages, shaped by ecological opportunities that had no direct precedent in the Permian, and developing along evolutionary trajectories that the extinction event itself had made possible by eliminating the organisms that would otherwise have occupied the available ecological space. Recovery, in this context, is best understood not as a return to a prior state, but as the construction of an entirely new biological order from the raw material of survival. The earliest measurable signs of ecological recovery in the marine realm appear in sections dating to approximately 2 to 4 million years after the extinction horizon, though the timing varies considerably between regions and between ecological systems. The first indicators are not increases in species diversity, which remained depressed for millions of years, but changes in the functional character of surviving communities. Biotubation, the mixing of seafloor sediment by burrowing organisms, begins to reappear in some early Triassic marine sections after its near-complete absence immediately above the boundary horizon, indicating the return of infaunal communities capable of processing sediment and oxygenating bottom substrates. The restoration of biotubation, while modest in its early expression, represents a functional recovery of a process central to marine biogeochemical cycling, one whose absence during the survival interval had allowed the laminated, anoxic sediment fabrics characteristic of the immediate post-extinction ocean to persist. The diversification of ammonoid cephalopods across the early and middle Triassic represents one of the most rapid and well-documented evolutionary radiations in the post-extinction marine record. From the severely bottlenecked survivor lineages that crossed the Permian boundary, ammonoid diversity expanded substantially within the first few million years of the Triassic, producing new genera and families at rates that reflect the combination of available ecological space, reduced competition, and the intrinsic evolutionary capacity of a lineage with high reproductive output and short generation times. By the Middle Triassic, ammonoid diversity had recovered to levels approaching late Permian values in terms of genus-level richness, though the ecological roles and morphological disparity of the new fauna differed significantly from those of their Permian predecessors, reflecting divergent evolutionary responses to a structurally different ocean. Marine reptiles, a group with no significant ecological presence in the late Permian Ocean, diversified rapidly into marine environments during the Triassic, exploiting the predatory niches vacated by the extinction of large Permian marine predators. Ichthyosaurs, whose earliest representatives appear in the fossil record within the first few million years of the Triassic, evolved from terrestrial or semi-aquatic ancestors and rapidly developed a fully aquatic body plan, convergently similar to that of modern dolphins, streamlined, with paddle-like limbs and a high aspect ratio tail fin adapted for sustained pursuit swimming. By the Middle Triassic, some ichthyosaur lineages had achieved body lengths exceeding 10 meters, positioning them as apex predators in a marine ecosystem that had, within a geologically brief interval, rebuilt a functional large predator guild from lineages that had played no such role in the Permian Ocean. The speed of this ecological transition reflects the magnitude of the opportunity created by the extinction, a vast productive ocean largely emptied of the predatory lineages that had previously occupied its upper trophic levels. Plesiosaurs, nothosaurs, and other marine reptile groups similarly diversified across the Triassic, collectively filling a range of marine predatory roles across different depth ranges, prey sizes, and hunting strategies. This diversification, multiple independent lineages of terrestrial reptiles, converging on aquatic and marine lifestyles within a few million years of the extinction, represents one of the most dramatic examples of adaptive radiation, driven by ecological opportunity in the vertebrate fossil record. The Triassic Ocean, emptied of its Paleozoic predator fauna, presented an array of unoccupied ecological niches that multiple independently evolving lineages exploited in parallel, producing a marine predator guild structurally analogous to that of the Permian, but composed of entirely different evolutionary components. On land, the recovery of terrestrial ecosystems proceeded through a sequence of changes in both plant community composition and vertebrate fauna that is documented, with varying resolution, in continental deposits from multiple regions. The transition from fern-dominated earliest Triassic vegetation to gymnosperm-dominated forests, more structurally complex than those of the survival interval, is recorded in palynological sequences from Middle Triassic deposits in Europe, Asia, and the Southern Hemisphere. The returning gymnosperm communities were not identical in composition to those of the late Permian, seed fern, and Corditalian lineages that had dominated. Permian forests were not restored, having been eliminated or severely reduced at the boundary, and their ecological roles were filled by conifer lineages and cycad relatives that diversified across the Triassic. The forests that developed across the Middle and Late Triassic were structurally recognizable as forests, with tall canopy trees and understory vegetation and complex soil communities. But their species composition and ecological history were entirely new. The diversification of archosaurs, the reptilian lineage that includes the ancestors of both crocodilians and dinosaurs, across the Middle and Late Triassic, represents the most consequential terrestrial evolutionary radiation of the recovery interval in terms of its long-term impact on the history of life. Archosaurs were present in the earliest Triassic as minor components of the survivor fauna, but their diversification accelerated through the Triassic as the ecological simplification of the earliest recovery interval gave way to more complex community structures, offering a broader range of exploitable niches. By the late Triassic, archosaurs had diversified into a range of body sizes and ecological roles: large herbivores, mid-sized omnivores, and active cursorial predators that positioned them as the dominant large-bodied vertebrates across much of the continental landmass. The first unambiguous dinosaurs appear in the fossil record in deposits dating to approximately 230 to 235 million years ago, roughly 20 million years after the Permian-Triassic boundary. Their initial ecological role was modest. Early dinosaurs were not immediately dominant, but were components of diverse archosaur communities in which multiple lineages competed for similar ecological positions. Their subsequent rise to dominance across the late Triassic and into the Jurassic was facilitated, in part, by a further extinction event at the Triassic-Jurassic boundary approximately 201 million years ago, itself associated with large igneous province volcanism that eliminated many of the non-dinosaurian archosaur lineages with which early dinosaurs had shared the landscape. The trajectory from the end Permian survivor fauna to the dinosaur-dominated world of the Jurassic thus involved not a single evolutionary transition, but a sequence of extinction and radiation events, each reshaping the competitive landscape in ways that progressively favored the lineages that would define the Mesozoic. The mammalian lineage descended from the cynodont therapsids that had survived the Permian extinction as small, physiologically advanced insectivores, diversified modestly across the Triassic, producing the first true mammals in the late Triassic in deposits from multiple continents. These earliest mammals were small, most estimated at body masses below 100 grams, and occupied insectivorous and omnivorous ecological roles in the understory and forest floor environments of late Triassic ecosystems. Their small size and nocturnal habitats characteristics likely inherited from the selective pressures of surviving the end Permian extinction defined the mammalian ecological position for the entirety of the subsequent Mesozoic, constrained to small body sizes and marginal ecological roles by the presence of large-bodied dinosaurs. occupying the dominant terrestrial niches. Their eventual diversification into the full range of ecological roles they occupy today would not occur until the extinction of the non-avian dinosaurs 186 million years later. The recovery of reef ecosystems, among the most delayed of all post-extinction biological recoveries in the Triassic record, proceeded through a sequence of ecological experiments involving multiple potential framework builders before the scleractinian corals that would dominate all subsequent reef systems established their ecological dominance in the Middle and Late Triassic. The 8 to 10 million year reef gap following the Permian extinction reflects not merely the time required for new reef-building organisms to evolve, but the time required for ocean chemistry, particularly carbonate saturation states, to stabilize sufficiently to support sustained biogenic carbonate construction. The eventual return of reef ecosystems built by an entirely new evolutionary lineage unrelated to the Paleozoic reef builders they represent illustrates the degree to which post-extinction recovery involves not restoration but reinvention, the construction of functionally analogous systems from biologically novel components. The Triassic world that emerged from the ruins of the Permian was, by its conclusion approximately 201 million years ago, a biologically complex and ecologically structured planet once again. Its oceans populated by diverse marine reptiles, cephalopods, and fish, its continents covered by gymnosperm forests supporting the first dinosaurs and earliest mammals. The path from the impoverished survival fauna of the earliest Triassic to this recovered world had required approximately 50 million years of continuous evolutionary and ecological reconstruction, a timescale that places in perspective both the depth of the damage inflicted by the end Permian extinction and the extraordinary generative capacity of life to rebuild complexity from its most reduced expressions. What had been lost was not recoverable in any direct sense. The Paleozoic world, its therapsid predators, its brachiopod-dominated seafloors, its trilobite-populated reefs, was gone permanently. What replaced it was different in its composition, different in its ecological structure, and different in the evolutionary trajectories of its constituent lineages. But it was, unambiguably, alive, and in that aliveness, it carried forward the thread of biological continuity that the end Permian extinction had come closer to severing than any other event in the history of complex life on Earth. The extinction of the non-avian dinosaurs at the Cretaceous-Paleogene boundary, approximately 66 million years ago, occupies a singular position in public scientific consciousness. It is the most widely recognized mass extinction in the geological record, the most thoroughly studied in terms of its causal mechanism, and the most culturally embedded in human understanding of deep time and biological catastrophe. The asteroid impact that drove it, a bolide estimated at 10 to 15 kilometers in diameter, striking the Yucatan Peninsula and releasing energy equivalent to approximately 1 billion nuclear weapons, is documented with exceptional precision across global boundary sectors and represents one of the most clearly characterized single causal events in the geological record of any mass extinction. The end Cretaceous event eliminated approximately 75% of all species on Earth, including all non-avian dinosaurs, the marine reptiles, the pterosaurs, and the ammonoids, losses of genuine and irreversible biological significance. And yet, by every quantitative measure applied to the comparative analysis of extinction events in the Phanerozoic fossil record, the end Cretaceous event is not the largest. It is not the most severe in terms of proportional species loss. It is not the most prolonged in its ecological consequences, nor the most fundamentally disruptive to the trajectory of evolution on this planet. By all of these measures, the end Permian extinction, the Great Dying, stands in a category of its own, separated from all other mass extinction events in the geological record, by a margin that current data consistently confirm across multiple independent analytical frameworks. The comparative magnitude of the two events is most directly expressed in species-level extinction estimates. The end Cretaceous event, with approximately 75% species loss, produced a biological crisis of extraordinary severity by any historical standard. The end Permian event, with estimated species losses of 90 to 96% across marine communities and approximate 70% of terrestrial vertebrate species, exceeded this by a margin sufficient to place it in a qualitatively different category of biological impact. The difference between losing 3/4 of all species and losing 9/10 or more is not merely quantitative. It reflects the difference between a biological system severely damaged but retaining much of its foundational ecological structure, and one in which that structure was itself eliminated, in which the food webs, habitat frameworks, and biogeochemical cycling processes that sustain complex life were disrupted at their most fundamental level. The end Cretaceous extinction, despite its severity, left intact a substantial reservoir of biological diversity from which recovery proceeded with relative rapidity. Mammals, diverse in the Cretaceous across multiple ecological roles, including insectivores, omnivores, and small herbivores, survived in sufficient taxonomic and ecological breadth to initiate a rapid adaptive radiation across the Paleocene that, within 10 million years of the boundary, had produced most of the major mammalian orders recognizable today. Birds, the avian descendants of theropod dinosaurs, similarly survived in diverse lineages and diversified rapidly across the Paleogene into the ecological roles vacated by their non-avian relatives. Marine plankton communities, while severely disrupted at the boundary, showed measurable recovery within hundreds of thousands of years in some groups. The ecological architecture of the Cretaceous world was damaged, but it retained enough structural integrity to support accelerated recovery. The end Permian extinction left no comparable reservoir. The Paleozoic marine fauna, the brachiopods, bryozoans, crinoids, rugose corals, trilobites, and fusolinid foraminifera that had structured marine ecosystems for hundreds of millions of years, was eliminated or reduced to ecologically marginal remnants from which no recovery of their former ecological dominance would ever occur. The terrestrial, therapsid fauna that had dominated Permian land ecosystems, its apex predator guilds, its diverse herbivore assemblages, its ecologically specialized lineages refined across tens of millions of years of Permian evolution, was reduced to a handful of generalist survivors from which the reconstruction of complex terrestrial communities would require tens of millions of years. The recovery interval following the end Permian extinction, 4 to 9 million years of biological impoverishment, followed by tens of millions of years of gradual ecological reconstruction, has no equivalent in the post-Cretaceous record. The causal mechanisms of the two events differ in ways that illuminate why their biological consequences were so different in magnitude, despite both qualifying as major mass extinctions. The end Cretaceous event was initiated by a single, geologically instantaneous impact, catastrophic in its immediate effects, but not self-sustaining in the manner of a volcanic province. The impact winter it produced, while severe, lasted years to decades, rather than hundreds of thousands of years. The acid rain, toxic metal deposition, and atmospheric chemistry disruption associated with the impact were intense but brief by geological standards, allowing recovery of atmospheric and oceanic chemistry to begin within time scales accessible to biological response. The Chicxulub impactor delivered its energy in a pulse. The Siberian traps delivered theirs across an interval orders of magnitude longer, preventing the geochemical buffering and biological recovery that pulse-driven catastrophes eventually permit. The sustained nature of Siberian traps volcanism, operating across hundreds of thousands of years and interacting with the carbon stored in sedimentary sequences through contact metamorphism, produced a carbon cycle perturbation of a duration and cumulative magnitude that the end Cretaceous impact did not approach. Ocean warming, deoxygenation, and acidification during the end Permian event persisted for time scales sufficient to devastate not just individual populations but entire ecological guilds. and the evolutionary lineages representing them. The end Cretaceous ocean, while disrupted, retained its thermohaline circulation, its basic chemical buffering capacity, and sufficient oxygenation in intermediate and deep waters to support the rapid recovery of marine communities. The end Permian Ocean, by contrast, experienced a chemical restructuring sufficiently severe and sustained that the Paleozoic marine biological community, built across 300 million years, could not survive within it. The four other mass extinctions recognized in the geological record of the Phanerozoic, the late Ordovician, late Devonian, end Triassic, and end Cretaceous events, each produced significant and lasting biological changes, and each represents a genuine catastrophe in the history of life. None of them, however, approached the end Permian event in the combination of taxonomic breadth, ecological depth, and duration of biological impoverishment that characterizes the Great Dying. The late Ordovician event, driven by glaciation and sea level change, produced severe marine losses but left much of the Paleozoic marine fauna intact to recover and continue diversifying. The late Devonian event, complex in its causal structure and extended across several million years, significantly reduced marine diversity, but did not eliminate the foundational ecological architecture of Paleozoic seas. The end Triassic event, associated with the central Atlantic magmatic province eruptions, produced substantial losses, but from a biosphere that had already been significantly amplified by the end Permian event and had not yet fully recovered from it. The significance of the end Permian extinction extends beyond its raw magnitude. It represents the only mass extinction in the Phanerozoic record that came genuinely close to eliminating the entire complex animal biosphere, not merely reducing its diversity, but threatening the continuity of the evolutionary process itself. Current estimates of marine species survival suggest that as few as 4 to 10% of marine species crossed the Permian-Triassic boundary. Had that percentage been lower, had the thermal extremes been more severe, the duration of oxygen depletion longer, the recovery interval extended by even a few additional million years of volcanic forcing, the survivor pool from which Triassic recovery proceeded might have been insufficient to rebuild the ecological complexity required to sustain diverse animal life. The margin by which complex life continued was, by the evidence of the geological record, narrow. What the end Permian extinction ultimately demonstrates, with a precision available from no other event in the history of life, is the relationship between stability of the physical environment and the continuity of biological complexity. The Paleozoic biosphere, its reefs, its forests, its diverse predator-prey networks and biogeochemical cycles, was not fragile. It had survived 4 previous mass extinctions, multiple glaciations, and hundreds of millions of years of environmental variation. Its destruction required a forcing agent of exceptional magnitude, duration, and chemical diversity, a large igneous province of interacting with volatile-rich sedimentary sequences, sustaining atmospheric and oceanic disruption across timescales that exceeded biological response capacity at every level, from the cellular to the ecological. The geological record preserves this event with sufficient resolution to reconstruct its mechanisms, quantify its magnitude, and trace its consequences through the subsequent history of life on Earth. That record describes, without exaggeration or interpretive embellishment, the closest approach to biological annihilation that complex animal life has experienced in the 540 million years since the Cambrian explosion first populated the planet with diverse multicellular organisms. Life continued, but the world in which it continued was irreversibly different, shaped at its foundation by the loss of what the Paleozoic had built, and by the characteristics of the few lineages that survived to build what came next.